a little bit to the FDA. And then just kind of some of the exciting things that I think CERSIs allow us to do.
Absolutely, that would be perfect. So I'll wait till 12:35 and then I'll introduce you and we'll get started.
Great.
All right. Well, welcome everybody. I'm sure more folks will trickle in, but it's time for us to start. This is the Yale-Mayo Clinic CERSI Scholar Virtual Research Seminar. For just as a welcome, and for those of you who are unaware, the Yale-Mayo Clinic CERSI is a joint effort between Yale and the Mayo Clinic and our faculty, working with the FDA to conduct research to address key gaps in knowledge, develop tools to support regulatory decision-making and the overall mission of the FDA. It's a collaborative research grant, and within the Yale-Mayo Clinic CERSI umbrella, we have 36 projects in various stages of completion, a wide range of work that crosses many disciplines from drugs and biologics to medical devices and generic drugs, and in a wide array of disciplines, which is really very fun. It's from survey research and lit reviews to clinical trials, and a lot of observational research where we leverage large existing data sources to try to advance science and increase our understanding about medical products and their use in the real world. The CERSI Scholar Seminar is a capstone event that we host every year. We've been a CERSI for, this is our 8th year now, and each year we've provided opportunities for students, postdocs, fellows, junior faculty who are doing and engaged in regulatory science research on our 2 campuses to present some of the work that they're doing and to get some feedback over the course of the year, and they earn a small stipend in addition to that. Probably 3 or 4 years ago, not surprisingly, around the COVID pandemic, we went from a monthly seminar to a one-day, sort of a half-day seminar where we invited all of our scholars to present over a full day. Everyone could be able to hear and listen to the work that's going on. And I think a year after that, we started reaching out and including scholars who are at the other CERSI sites, including Johns Hopkins, the University of Maryland, and at the Stanford and UCSF joint CERSI, and now from the Triangle CERSI, the newest CERSI. And we've always reached out and included folks from the Harvard Regulatory Science Center as well. So we're really delighted to get started today. We have our guest speaker, Dr. Paul Kluetz, who's a medical oncologist and the Deputy Director of the Oncology Center of Excellence at the FDA. In addition to contributing to the strategic leadership and operational oversight of the Oncology Center of Excellence, he holds a leadership role in the Office of the Oncologic Drugs, overseeing the regulatory review of solid tumor drug indications within CDER. Dr. Kluetz founded and leads several initiatives, including creation and direction of OCE's patient-focused drug development program, its real-world evidence program, trial modernization efforts, including pragmatic and decentralized trial designs, use of digital health technology, the OCE's oncology labeling program, and support of the OCE's extramural research portfolio. He has published over 150 peer-reviewed manuscripts and remains clinically active, caring for patients and supervising medical residents at Georgetown University Hospital. And I can say I've had a great opportunity to work and get to know Paul over the years through one of our CERSI, or 2 of our CERSI projects now, including one of the clinical studies we've been engaged in. It's really just so delighted and happy to have you, Paul, to kick off our event.
Thanks, Joe. And it's good to see also some familiar faces. I've seen worked with Dr. Alexander a bit before, Carrie Gross I see too. So I'm excited to see both of you guys. And I'm also really excited to speak to the future of therapeutic drug development and biology and whatever, public health, all the things that these junior scientists are interested in. I'll touch on some of these topics, but really what I want to do is instill my love for teaching and for learning and for evaluating complex problems And for us, these are problems that have real impact to the public health. One of the things that I really enjoy about the CERSI mechanism is it's very applied. You know, we're trying to identify projects that can be really quickly applied into actual regulatory review of products that we regulate. So, you know, these are really impactful studies. So I'm gonna share my screen real quick so I can have some slides for you as well. Let me know if I've done this correctly. I think I'm getting better at it. Can you guys see my slides? Yep. Okay. So again, thanks for the kind introduction, Joe. My name is Paul Kluetz. I'm a medical oncologist. I trained at the National Cancer Institute and was very interested in GU oncology and prostate cancer research earlier. Came to the FDA after fellowship. Really thought I was just going to be here for 2 years to learn how drug regulation works, and then move off and work at Yale, at Hopkins, at Georgetown, somewhere, and be a translational clinician because I really love to see patients. But I found it to be fascinating, and I hope some of you at some point in your career may want to come over and work at the Food and Drug Administration. So I put this slide up with a quarter just to give you a sense of the breadth of the products that we regulate in the United States at the FDA. A lot of people don't realize all that we do. It's about 20 to 25 cents of each dollar spent in the United States is on products regulated by the FDA. It's not just drugs and biologics. It's not just devices. It's not just food. It's tobacco, radiation products, veterinary products, cosmetics. It's quite a bit. What I do is any product that is intended to diagnose or treat patients with cancer. And so it spans the product centers a little bit, and I'll talk about that. The one bullet that I love that is probably my favorite part of my job is this science and collaboration piece. It's actually a part of the mission of the FDA. We're looking to create a consistent regulatory environment for product development, but we're also, you know, interested in innovating and helping to develop products in an expedited way through endpoint development, through trying to look at different sources of evidence, et cetera. And I'll talk about that a bit. Two things that are very commonly misconstrued is we don't actually regulate the cost of products. A lot of challenge right now in the US is the cost of healthcare in general, quite frankly, but certainly drug prices are at the forefront of policy questions right now. Some of our most amazing biologics are being priced, you know, in the $500 million, $2 million range. So we have, you know, some ways to go with trying to understand pricing. We don't know how much anything is going to cost as we're reviewing it. We are solely focused on the safety and the efficacy of the product and prior to marketing, and then how the price is set is out of our control. We also don't regulate the practice of medicine. So that's where off-label use comes from. We approve a drug in prostate cancer, let's say. That drug can be prescribed by a prescribing physician for any other indication they feel makes sense for the patient that's in front of them. It may not get paid for, but we don't regulate how doctors practice medicine. We do regulate how companies market their drugs and biologics, and a drug company can't market their drug for something that it's not indicated for. So I mentioned briefly that our role is sort of unique in the FDA. We started this inter-center institute called the Oncology Center of Excellence by congressional mandate now 6 years ago or so, based on the fact that oncology product development in general is extraordinarily robust right now. I think it's not because oncology regulators are so great or medical oncologists are so great. It's actually because we've spent billions of dollars of basic science research over the last 10, 20 years to understand the biologic and immunologic underpinnings of the disease. And so we're starting to really harvest the benefits of that understanding, and we've seen rapid progress in many products. Not only do we have lots of drugs, which is regulated in the Center for Drugs, which is CDER. We have lots of biologic therapies, vaccines, T-cell therapies, and those are regulated in the Center for Biologics, or CBER. And then we have devices, therapeutic devices that ablate tumors, but also in vitro diagnostic tests that select patients that are most likely to respond, and that's in the Center for Devices. And if you think about oncology and products that have been recently developed, they're spanning all three of these product centers often. And so the intent was, rather than a center that's focused on a product, let's have a center focused on the disease so that we can share a common understanding and depth of clinical trial design and experience across the clinical development of centers. And so that's what we do, the Oncology Center of Excellence. I will say that my job is interesting insofar as it's a significant balancing act, and I have to balance things every single day. On the x-axis is probably the biggest balancing act. That is, we would love to have complete certainty that a drug is effective and that it is safe for as long as it's going to take, and have precision around what the safety of these drugs are. But with increased certainty comes increased data, increased number of trials, increased time, and that's called increased regulatory burden to sponsors. And we will be hearing that we're too cautious, we're stifling innovation, and there's too much regulatory burden, and will be pushed to the left. On the other hand, if you overdo it with expedited programs, you reduce the number of trials, you reduce the amount of data that you receive, you're clearly going to reduce your certainty. And this is a safety example. Maybe you'll miss a low incidence, high severity adverse event and will be called asleep at the wheel, toxic deaths. Or maybe you have less certainty around the effect that your drug purports, efficacy. So we need to block that balance. I personally feel like in oncology, we're reasonably in the middle, but we're always recalibrating. And on the y-axis is kind of how do we interact and how do we operate from a regulatory perspective? We have to be flexible because every product is new and different, and their clinical development is different. But we have to be consistent in our decision making, because there's companies that are trying to create a product with a 5 or 10 year time horizon and amount of money that they have to spend. So they need to kind of understand what we've done before and feel confident that it'll be done the same way in subsequent approvals. We have to be efficient with our reviews, but also very thorough. And of course, we have to be completely independent in our decision making, and yet we have to interact with people in the clinical trial environment, or we're going to be asking for things that are impossible or impractical. So, all of this is a balancing act, and I think you have to have a bit of a thick skin to work here. So, where does science come in? Well, I mean, in oncology, we are kind of in a very different area now than we were when I first started in 2010. So clinical trials, as I said, have been very successful over the last several decades, and we have scores of novel drugs and biologics to treat patients with cancer across many different diseases, much more busy and much more options. And that is very good and creates some challenges as well. So when I started in 2010, there was like 2 or 3 prostate cancer drugs. We had mostly cytotoxic chemotherapy. That's the chemotherapy where you kill rapidly dividing cells indiscriminately. And where are rapidly dividing cells? Hair, the mucosa. So you get sores in your mouth, you lose your hair. This is the classic chemotherapy for cancer. There were not many available therapies across almost all diseases. Most diseases in metastatic solid tumors like melanoma, lung cancer had a very grim prognosis. The early clinical information, in other words, how often we were shrinking tumors, was pretty modest. So 10, 20% of patients, 30% of patients were having responses. And we didn't have a lot of digital health technology. It was becoming available. The World Wide Web was becoming available. Smartphones were becoming available, but it was nowhere near the information load that we have now in today's era. So the clinical landscape, treatment landscape is very paternalistic. You have this, I think you should take this. Or you have this, I'd like to put you on this clinical trial and you will be on this arm. And that's just kind of how it was done. And patients didn't have a lot of information to access to be informed. Today, many therapies, many available therapies, very early on, we see significant anti-tumor activity. So whereas 20% of the patients might have responded, now we have 70% of patients responding, or 60% deep, cancer almost going away, and for a more prolonged period of time. And I think we have a lot more therapies and a lot more information. And so now the clinical landscape is shared decision-making, and patients have a lot of information, some true and some not true, unfortunately. And that's a good thing, but it's creating some issues that we haven't really seen. So there's accrual challenges. So when we have clinical trials, there's lots of clinical trial options now, and many of them are even in the same disease and very similar drugs competing against each other. We have much smaller US patient subpopulations because a lot of these are being offshored. Trials are offshored to different countries. And because of that, we have a lack of representativeness. So there's a big push for more diversity in age, ethnicity, race, all the things. We want the data to look a lot more like the people that we're going to be treating in the United States. Even if you accrue a patient on a trial, we're having issues now with retention of patients on trials, especially on control arms. You get randomized to the wonder drug because the information age is spewing out all the information of how great it is. And if you get randomized to the control arm and you know that you're on that, patients are leaving the trial. And that is creating some major challenges, which we've talked about in advisory committee meetings. Then, of course, there's always the question of cost and resource constraints. Trials are expensive, and the push is, less patients, quicker, less number of trials. All of these are pressures that are driven in some ways by cost constraints. And in general, it is burdensome for patients to be on trials as they're currently constructed. So all this to say, there's a lot of regulatory science that needs to happen to understand how can we modernize how we generate evidence, how can we make trials cheaper, more efficient, more patient-centric? Can we develop endpoints that we can see information earlier in some diseases, et cetera? And this is where all of you come in. There's so many questions that we have to answer with new drugs, and the FDA approval is just the very beginning. Now we put another new drug in a space that has 4 or 5 existing drugs. Is the new treatment better than the existing treatments? Because FDA doesn't actually have a comparative efficacy requirement. The drug just needs to be safe and effective. It doesn't need to be more effective than the existing treatment for regular approval. So there's comparative efficacy questions. There's sequencing questions. Which drug should you put first, second, third? Is it equally effective if we lower the dose or lower the duration of a therapy, so de-escalation? More therapy in high-risk groups question. We're very interested in all those subgroups that I just talked about are underrepresented, right? So I want to know about older adults. I want to know about different racial ethnic subgroups, organ dysfunction, what happens if someone takes this drug with moderate hepatic injury. Those are questions that we need to answer. And increasingly, we'd love to see doses of cancer drugs that are more tolerable to patients. So better understanding of more tolerable and optimal doses. And none of these questions are going to be helped by evidence that's not high level, right? So one of the things that's slightly, I don't understand as much is, well, we're making a payment decision. So for payers, we don't really need, the kind of data that FDA needs. We can accept a real world observational study, let's say. But, if you're making a decision on whether to pay for a drug, that's probably as important for access of that drug to patients as the decision to approve it in the first place. Or sequencing, or clinical trial guidelines like NCCN. All of these require really, really high quality evidence. And so we shouldn't accept lower levels of evidence for some of these questions. They're all really important questions. So when I think about where can we start to do better and modernize how we look at evidence, or I guess develop evidence, these are all areas where you might start to see some questions that you all might be able to start to try to answer. So there's lots about protocol design. So for the clinicians, the MDs in the audience, trial designs, creating platform trials or master protocols where you can pull drugs in and out in a much more efficient way is something that's very interesting. For the statisticians in the audience, we really are interested in adaptive design. So rather than serially running the early phase trial, start up a whole new trial, the new protocol for the phase 2, stop, design a randomized phase 3. That was how we used to do it. Now we like to, it's great to have integrated phase 2, 3 trials where you're staying in the same trial and then expanding to randomization. Using Bayesian statistics or other statistical methodologies are areas of exploration. And then this decentralized clinical trial situation, which is what we had to do in COVID. So anyone who's a clinician working at Yale knows that we pretty much treated a lot of patients outside of the clinic and they stayed home a lot more. And that is attractive to reducing some of those challenges. As I said, patients dropping off of trials. So you can make trials more efficient and have them conducted closer to where patients live. And actually you can pretty much have the trials somewhat assessing the same things as they always have. But you could also just ask less and focus more on the objective of the trial. So this is like so-called pragmatic clinical trials where it's a little bit, there's less complexity in the trial itself. It's more like randomizing patients and then letting them be treated as they normally would in standard of care on two different treatments and everything in between. There's various levels of pragmatism. That's an area that we think is important. And then an area that has got a lot of research right now within our programs is looking at how we can better use the source of the data. So most of the data source in cancer clinical trials has always been case report forms. In other words, very detailed, manually curated forms that are filled out by clinical research staff telling how the patients have done. It's very labor intensive. Can we pull more information straight out of the electronic health records to, begin to fill these things out without human beings? Can we actually put wearable sensors on patients? Can we have them answer electronic patient-reported outcomes and import that structured data straight into data sets? So a lot of this is I think, an area that we should be investigating. And of course, within data sources is this idea of real-world data and real-world evidence and observational studies. That's an area that we have a lot of work in as well. So what are we doing as far as the scientific component of this? And I will say all of these things we're seeing on the regulatory review side, believe me. So we have large commercial sponsors proposing decentralized trials, pragmatic trials, real world evidence. So we're seeing it come in, but we'd like to understand it at a very detailed level. And in order to do so, we were running a lot of our own studies and funding people like CERSI to help us with some of this. So our scientific collaborative, which is led by Julie Schneider, who I think is on the call, is really intending to support our staff's ability to plan and conduct high quality applied research efforts in collaboration with really smart external scientists. And that is one of the best parts of CERSI, is that we can be very interactive, as I think Joe and Carrie can attest for one of them that I'm involved in. Our scientific collaborative program is very, very busy. We put out a lot of publications. We have a lot of internal educational events and public workshops, and they're all informed in part by some of the funded research that we do. So these are some of the buckets of areas of study that we fund through CERSI specifically. We look at health equity questions. We are looking at immuno-oncology and precision oncology questions, biomarkers. We're looking at safety. We're looking at what I call patient-focused drug development. A lot of that has to do with measuring symptoms and function of patients. In cancer research, this has been an interest for decades, hasn't been done very well normally, and we're starting to see a lot more interest in it. Why? Because there's so many drugs. It's becoming very attractive to have an equally effective drug that's way more tolerable. How do you know it's more tolerable? You got to know what symptoms it's causing, and you got to know how people are functioning and carrying out their daily activities. And that's about patient-reported outcomes and wearable devices. Precision oncology is biomarker development. I talked about real-world data. We're looking at all sorts of real-world data sources. We have a lot of research collaborative agreements with groups like Flatiron, CODA, and those people that are curating large data sets. And then a lot of that statistical methodology and trial designs and endpoints, decentralized trials, that's that bucket. We did a paper, or Julie wrote a paper not that long ago, just to show that actually one of the metrics that we think is interesting is that our publications are highly cited. And so we're making an impact. And we're not only learning ourselves, but we're helping stimulate a little bit, of the external research too, which is, I think, great. Just to end, I'll just give you like an actual example, which is the one that I'm directly involved in. I said a little bit about why I think it's important to measure symptoms and function for patients with cancer. Everybody knows cancer therapies can be difficult to take. You're not going to have a blood pressure medicine that causes mucositis and your nail bed problems and ocular toxicity. Only in oncology are we somewhat okay with that level of toxicity, because the disease itself is so devastating. But we can do better. And if you can find a more tolerable treatment that's equally effective, I promise you patients will want to take it more. But how do we measure it? So these are areas of investigation. Electronically captured patient-reported outcomes is probably the closest to regulatory use right now, but we're very interested in wearable devices and sensors as well. These are different modalities of how you can obtain symptom and functional data. So clinical outcome assessment is the name of the broad category that FDA uses. And within those, and this is just how you measure symptoms or function, clinician can actually grade it. That's called a clinician-reported outcome. An observer can look at it and grade it. So a parent observing the number of vomiting episodes that a child has, let's say, who can't report for themselves. Patient-reported outcomes are the surveys, the validated questionnaires that are asked. Performance outcomes are like the 6-minute walk test. So you ask someone to do a task and you measure it. And then in oncology, what we have done through our patient-focused drug development program is to try to organize at least the commercial sponsors around being a little bit more focused with what we want to see every time. Rather than this sponsor wants to look at social well-being, this sponsor wants to look at quality of life more broadly, this person wants to look at fatigue only, to say, look, we know that there's a very narrow set of symptom and functional outcomes that are important to patients, that are sensitive to the intervention we're trying to study, and that therefore could inform what we do, regulatory decisions. And the core outcomes that we have is disease symptoms, symptomatic toxicities, your overall side effect bother, and how you're doing physically, your physical function, and whether you're able to work and carry out your daily activities. So these are what we're asking for in every trial, and it's been remarkable at how it's been picked up, and we're now starting to see more consistency. So when we looked at tolerability, there was a working group led by Friends of Cancer Research that made it very clear as we talked to patients that, you know, the International Conference for Harmonization's tolerability difference or definition was really about just the degree that patients could handle it. The degree to which patients can handle overt adverse effects. So it's very built on kind of a cytotoxic chemotherapy. You're tolerating it if you're still taking it. It's kind of and the patient saw it and said, you know, it's more than that. You know, a grade 1 or 2 low-grade rash over time is going to make me stop that therapy, and yet physicians have been saying this is tolerable. So we've kind of got a broader definition where it's symptomatic toxicities and non-symptomatic toxicities. And it affects the ability, but also the desire of the patient to stay on that therapy. Because anti-cancer agents are actually oral now too. So it's no longer like they go to the infusion unit and they don't have a choice. They have the choice every day to take the pill or not. And we are seeing adherence problems. So that brings me to how can we do better with how we assess these? And this is a really exciting study that we put together, I don't know, 3 years ago or so now, which was pretty, it's the first large study that we funded, clinical study. So this has over 200 patients being treated with standard cytotoxic chemotherapies that are pretty significant chemotherapy burden. So I wanted to look at a patient group that was going to have a functional decline and then a recovery so that we could see some range of functional ability. And then I want to measure function across modalities, including wearable devices. So patient-reported outcomes, these are all the clinical outcome assessment modalities we just talked about, right? The clinicians are going to grade it. The patients are going to grade it with their patient-reported outcome. We're going to ask them to do a walk test, and we're going to measure that performance outcome. And we're going to put a wearable device, a Fitbit, on them and see what their activity levels are. And we're going to watch all those modalities over time, and we're going to try to understand what's more sensitive, what's the meaningful change in a Fitbit compared to a PRO. And the study was successful. Keynote Address was amazing, and all of the clinical investigators were really, really dedicated to this study. So we're awaiting follow-up. I'm going to meet the group at ASCO actually and talk with them a little bit about what the first kind of analytic studies will do. So with that, I will end by saying I love what I do. I love this part of what I do specifically. I'm really enthusiastic that Joe has set up this for me to meet you all. I'm happy to answer some questions. And we'll continue to use the CERSI mechanism more broadly, I think, over the years.
Thanks, Paul. That was terrific. We have a couple minutes for questions if people want to unmute. just to get started, I do have one, which I think is one of the most interesting things that is facing FDA these days, which is the sort of interesting competing demands that are happening with real world data, so to speak, while also being kind of, well, for lack of a better term, kind of better in the endpoints that we're focused on, like these patient reported outcomes and whatnot. Because the challenge, I think, in oncology, many of the traditional endpoints that have been used over the years to secure product approval, endpoints like response rate, progression-free survival, they're actually not, quote unquote, in real world data, right? You can't get them from there. So it creates this really valuable opportunity to kind of like change the rules, so to speak, and think about like, well, what data do we need in these larger data sets that we're going to be using for monitoring, surveillance, or perhaps supplemental indication approvals? How do we get these patient-reported outcomes into the data so that the data can help us have the insights that we want? I'm curious, just sort of like what you've seen, or some exciting things that are kind of on the horizon doing exactly this.
Yeah, one of the things that I've seen over the years, and this is not going to be, some people will hate this, but the best endpoint we have in oncology is response rate. It's the one endpoint that I absolutely feel very confident is being driven by the effect of the drug and not some other confounding influence. And I also think if it's a deep response and it lasts for a significant period of time, that it has, some face validity of benefit, acknowledging that the toxicity needs to be, thought of within that, and that can be a challenge. That endpoint doesn't exist in the real world, right, Joe? So the best endpoint we have doesn't. Then all of our time to event endpoints, which are just for the early career scientists, one of the challenges, the vexing challenges of almost every therapeutic area, is if you have a time to event endpoint, meaning you're waiting for something to happen that's driven by the disease, whether it's functional decline in Duchenne's muscular dystrophy, whether it is the time it takes for the tumor to progress, whether it's the time it takes for someone to recur in cancer, that cannot be really well evaluated in a single arm trial. And so you have to randomize. So the best real world endpoints that I've seen are time to event endpoints. And yet we don't have a lot of randomization. We don't have randomization in an observational study. So thus, Joe, I think why the pragmatic trial thought has become much more exciting, because you can maintain randomization and then just deploy it and start to look at real world endpoints that may actually be more clinically meaningful. So for instance, some of them are time to treatment discontinuation. I'm speaking only about oncology right now, but time to treatment discontinuation. Okay. So that event, treatment discontinuation, could be because the tumor progressed, could be because the patient couldn't handle the toxicity any longer. But in any event, they're not taking the therapy for not a good reason. So it's a bad event and it's a real world event. I know, I think survival obviously is a great real world. It's our best real world endpoint. It's a time to event endpoint, right? So, and I know Caleb Alexander's on the call, so we could chat at length on how people want to start to look at single arm cohorts with time to event endpoints with an external control. That is extraordinarily challenging to do well. So I feel that tension every day, Joe, to tell you the truth. And we did do a very kind of interesting study with Friends of Cancer Research, where we took like 6 of the biggest data purveyors right now and asked them to come up with some kind of definition for real world response, and then look at how their different databases kind of matched up with what they were getting from a common lung cancer treatment. Pretty consistent. Imaging assessment frequency was actually relatively consistent. So there was some interesting insights, but there's a lot of work to be done there, I think.
Great. Since we have you, we'll do at least one, maybe 2 more questions. Sten Vermun, the former dean of the Yale School of Public Health, I see your hand is up.
Yes, thanks. That was a terrific review, and we really appreciate your joining us today. I had a question about how to measure the metrics of impact. You mentioned that you were being cited twice as often as the PubMed average, which sounds plausible that that's higher impact. But fields like oncology and cardiovascular disease are so vastly larger than many other fields, endocrinology, rheumatology, infectious disease, whatever, that one might expect these to be cited more frequently given the size of the field.
Yeah.
So would you speculate a little more about how we can actually measure impact of the kind of science that we're putting out there?
Great question. Julie Schneider is on the call, and I don't know, Julie, if you've stepped away or not, but Julie wrote the paper, and I don't know how they did that, and I'm not an author. so I don't know how they did the
Well,
relative.
I probably should read the paper and try to get the answer to my question. Thank you.
No, but I think Julie's on, but I don't know if you corrected it for the relative number of sort of cardiovascular and oncology.
We worked with a group at NIH who is really expert in doing these citation analyses, and I'm forgetting, it was a standardized measure that they recommended, and the name of it I'm forgetting right now, but I could definitely follow up with you. I mean, it's in the paper, which is a little bit old, but it's definitely like it was a measure that they recommended that I believe corrects for those types of issues. But I could follow up with you and provide you more information.
Julie, while I've got you, because Sten's other question was interesting. What other metrics can we use as a community to?
That's such a great question. I mean, we really struggle with that because, I mean, even citation analysis is so limited, right? Like in that paper, we also like tried to look at citations in like clinical practice guidelines and in clinical trials themselves, but it's so challenging, and that's why we had actually worked with this group at NIGMS that really kind of does this, because of course NIH is always really trying to figure out what the impact of their work is too. But it's a really challenging question, and the farther and farther you get away from the research, the harder it is to measure. So I'm not sure I have a good answer for that, but we try. So
I don't think there is a good answer. I'll point out that ORSI, the Office of Regulatory Science and Innovation, makes all the CERSIs. How are our projects had a measure our impact, and it's always like we know how it's being understood within the agency, where we know if it's been published, but impact is such a challenging thing to measure. We'll do one more question. I see Manun Shahzad. Your hand is up.
Yes, thank you. Thank you for a fabulous presentation. I had a quick question about sort of the representativeness slides that you had. It's been interesting because I think the FDA has had sort of a renewed focus on being on trials that are more representative. And it seems to me that representativeness is such a multifaceted concept, like you could be representative on many dimensions. And I was sort of wondering whether there are ways in which, is the FDA's idea sort of totally broad, like any type of improved representation is better, or are there sort of disease-specific ways to think about representation, or sort of how is that problem sort of made into something that's more actionable?
We have a huge problem with representation, and I look at it from 40,000 feet, and then I can get way down to like 5 feet. At 40,000 feet, we'd like to see U.S. patients, period, right? And we're not even, in some cases, I've seen a decent number of approvals that have had no U.S. patients, mostly multi-regional. Now, from a regulatory perspective, what we look at is, are the patients representative of or similar enough to the U.S. population to where we can, feel comfortable that there's nothing major. To your point about, do we look at specific populations in some ways a little different? Well, if it's a drug for multiple myeloma, and we know that Black patients with multiple myeloma have a unique either outcomes or some unique biology, then we feel even more strongly that we need more data on that population. So there's that sort of scientific biologic rationale. And then there's just this sort of societal, like just fair access component, like why isn't this happening? So I think a lot of the access issues is concerning outside of the sort of biologic plausibility of a subgroup doing differently than another subgroup. And then big picture is we have to make a decision. The difference between academia and my job is, people can talk in a room and say, this is terrible, we should do better, and what should we do? And then say, okay, let's just do another grant. For us, we have an application with a drug and a life-threatening disease that has a really solid outcome. And yet there's no representation of, probably 2 or 3 important subgroups. If we don't approve it for that reason, everybody loses, including the very subgroups that will not have access to it. And if we just keep approving it without saying anything, then it's not going to get better. So we're somewhere in between, trying to push to say, give us a plan. We want a representative U.S. population. How are you going to achieve that? And I hope over time, because this is a cultural shift that has to happen, we're going to start to see more representativeness across the board.
Paul, thank you again for making the time to be with us today and to kick off our CERSI Scholars seminar. You're more than welcome to stay on and join. I know you may have other tasks and responsibility ahead of you, but thank you for doing this. We all really appreciate it.
My pleasure. I got to run to another meeting, but it's great to see everybody, and good luck with all your research.
Great. And thank you, Julie, for jumping in too. We are gonna
sorry, Joe, I just did put some info in the chat about the metric that was used. Is called relative citation ratio. It was my colleagues at NIH who recommended this, so there's a paper there in case.
Oh, very cool. No, thanks.
Okay,
so now we can check it out. Yeah,
I hope that's helpful. Thank you so much. I'm gonna jump off too.
Okay,
bye.
So we're going to move to the first session of our seminar, and we've tried to do our best to kind of organize a little bit of like with like. It's very imperfect because we can't we didn't pre-specify what you guys were supposed to be working on or studying over the course of this year. So we have 12 presentations broken into 4 sessions. And the first one today is going to be presented by Haidong Liu. It's called Towards an Enhanced Framework, the 4 Targets Approach for Generating Real-World Evidence in Medications for Opioid Use Disorder. So take it away.
Great,
thank you, Haidong, for joining us.
Could you see my slides?
Yep.
Okay, that's great. Hello, everyone. So my name is Haidong. I'm a pharmacoepidemiologist working in the program in addiction medicine at Yale School of Medicine. So today I'm truly honored to be here and present my CERSI project on the 4 targets framework for generating real-world evidence in medication for opioid use disorder. So first, I will briefly talk about the motivation of this work. Opioid use disorder addiction continue to be a public health challenge in the United States and contributing to the ongoing overdose epidemic. So fortunately, we have 3 FDA-approved medications for opioid use disorder, the MOUDs. So especially, the study have shown that the methadone and buprenorphine are very effective in reducing opioid use, overdoses, and mortality. And so nowadays, we have increasing MOUD options and formulations. Moreover, the drug supply environment is constantly changing. That means there are still a lot of important questions on comparative effectiveness on MOUDs that need to be answered through high-quality research to inform policy and practice. So good news is that the increasing availability of routinely collected real-world data from electronic health records, insurance claims, and registries presents great opportunities for MOUD research. So therefore, the key is to develop a solid method framework to improve the quality of real-world evidence in MOUD research. So on the other hand, I found a series about epidemiologists. The epidemiologists love targets. So for example, we put a lot of emphasis on the concepts of target population, target trial elimination, and there's also a concept of target validity and target bias. So furthermore, we also learn to use target learning, maximum likelihood estimation for our analysis. And some of us also love the term target estimate and target parameter. So I'm thinking maybe we could synthesize the targeted concepts in the existing literature and then introduce a straightforward 4 targets conceptual framework, including target trial, target population, target estimate, target validity to guide the observational research to generate reliable and policy-relevant real-world evidence in MOUD research. So to place our 4 targets framework in context, I'm going to use an example of high dose versus standard dose buprenorphine as a treatment for opioid use disorder. Assume we want to address the following research question. What is the comparative effectiveness of receiving high dose buprenorphine versus receiving standard dose buprenorphine on retention treatment, hospitalizations, and mortality among people with opioid use disorder? And the data available to us is the electronic health records from the Veterans Health Administration, VHA. To illustrate our 4 target framework, we start with the concept of target trial emulation. That is, we utilize our observational data from the VHA to emulate a hypothetical pragmatic target trial. So briefly, the target trial emulation is an application of the design principles from randomized trials to observational research. The idea of the target trial emulation is not new and can be traced back to 70 years ago. So in 1953, the Haidong mentioned that we need to address a series of questions before initiating an observational study. So one important question is, how would the study be conducted if it were possible to do it by controlled experimentation? So in other words, it means we should have a target trial in our mind when we conduct an observational study. And then I also want to take the opportunity to acknowledge a great paper by several faculty from Yale School of Medicine published in 2000 in New England Journal of Medicine. So in this paper, the authors mentioned that a specific method used to strengthen observational studies adapts the principles of the design of randomized control trials to the design of observational studies, including identifying the time zero for determining the patient's eligibility criteria, then using the inclusion and exclusion criteria similar to randomized trials. And we also need to adjust for the difference in baseline prognostic factors to the outcome. And finally, using statistical methods such as intentional treat analysis that is similar to those of randomized control trials. So actually, over 20 years ago, in this phenomenal paper, some preliminary protocols for target trial emulation has been outlined. Finally, in 2016, in American Journal of Epidemiology, Hernán Robbins from Harvard proposed the target trial emulation framework and specified the components of the target trial protocols, including specifying the eligibility criteria and treatment strategies, treatment assignments, follow-up period, outcome, causal contrast of interest, such as intentional treat and per-protocol effects, and the analysis plan. So by following this approach, we could draft our target trial emulation protocols for our example on high dose versus low dose buprenorphine, as shown in this table. So our goal is to estimate the observational analogs of intentional treat and per-protocol effects from the VHA. One important note is that drafting the protocols of target trial emulation is an iterative process that involves collaborative efforts among the clinicians, statisticians, epidemiologists, and also the patient representativeness in order to address the most important questions through rigorous study design and analysis. And I also want to mention that, like any other observational analysis, target trial emulation can suffer from unmeasured confounding. So therefore, we often need to conduct some sensitivity analysis, such as the negative controls, instrumental variables, and quantitative bias analysis to check the robustness of our result to unmeasured confounding. So I would say the target trial emulation provides a useful framework for observational research to generate causally interpretable real-world evidence. And it's also structured reasoning and collaboration and enhance the transparency in reporting. So importantly, the target trial emulation can minimize the bias related to internal validity, such as the selection bias and immortal time bias. However, target trial emulation is not enough. Similar to randomized trials, the strict eligibility criteria is in target trial emulation can result in a highly selected study sample that is not representative of the target population of interest. So it limits the potential of the generalizability. So moreover, one often ignored point is that the clone sensor-based approach and sequential next trial design that are frequently employed in the target trial emulation to account for immortal time bias allow some study participants to be included in the study in the analysis multiple times, which makes the study sample difficult to define. So these issues exacerbate the fundamental question: to which population can the findings from targeted trial emulation be applied? So consequently, the role of the target population and the target estimate becomes prominent. So from target trial emulation, we obtain the intention treat and per protocol effect estimates in the study sample. So in our example of high dose versus low dose buprenorphine, the study sample is the veterans with opioid use disorder who received buprenorphine in the VA care. But the fact is, only 20 to 30% of people with opioid use disorder in the VA care received the medications. The vast majority of the people with opioid use disorder never received the medication treatment. But consider the one major objective of the MOLD research is to expand the treatment access and reduce disparities in medication receipts. It becomes evident that solely focusing on the internal validity in the study sample is not sufficient. So specifically, in our example, we are more interested in the treatment effect among all the people in the VA care who were diagnosed with opioid disorder, regardless of their treatment status. So correspondingly, our target estimate are the intentional treat and per protocol effects in the target population. So that is the treatment effect among all treatment eligible people with opioid use disorder in the VA care. So if the study sample is a random sample of the target population of interest, then the results from target trial emulation can be directly generalizable to the target population. However, oftentimes the study sample is a non-random sample of the target population. So in our example, those received the treatment for opioid use disorder and those never received treatment may differ by social demographic characteristics such as age, race, and socioeconomic status. And they may also differ by some health conditions such as psychiatric disorders and pain conditions.
Haidong, I just want to make sure it's been 10 minutes just to.
Oh, okay. Yeah, I think I only have 2 or 3 slides.
Okay, great.
Yes. So different distribution of the participants' characteristics contribute to the effect heterogeneity between study sample and target population. So fortunately, we have some statistical methods such as inverse probability, outcome regression, and double robust estimators that can be used to quantitative generalize the results from a study sample to a specified target population. So finally, we move on to the target validity. The target validity is a joint measure of the validity of the causal effect estimate with respect to the specified target population, which incorporates internal validity and external validity into causal evaluation. So it's a summary. So emulates and target trial can enhance the internal validity quantitatively generalize the study results to a specified target population can improve the external validity. By doing so, we are able to achieve reliable and policy or practice relevant effect estimates in terms of target estimate and thereby improve the target validity. So this slide shows the conceptual scheme of our 4-target framework, which could guide the generation of real-world evidence for MOUD research and beyond. So finally, I'm thankful for the support from my mentors, including Greg Gonzalez, David Falin, and Fanny. And I'm also grateful to the my brilliant collaborators, including Carol Rudolph, Michael Hahe, David Fink, Chris Ranch, and Catherine Lesko. Thank you for your attention. Sorry, I just
Haidong, that was great. Very, very interesting. We're going to hold questions to the end of the session for the 3 presenters who are going. So please hang tight. Our next presentation is from Tamara Hughes from the University of North Carolina, who's going to be talking about enhancing the role of pharmacies and reduce opioid misuse and abuse in racial and ethnic minority and rural communities. Take it away, Tamara. Thank you for joining us. Welcome.
Thank you. Let me pull up to share my screen. Okay, let me presenter view. All right, able to see my slides perfectly?
Yep.
Okay, that sounds good. Okay. Good afternoon, everyone. My name is Tamara, and I'm a postdoc at the UNC Eshelman School of Pharmacy, and really excited to present to you today on the topic of enhancing the role of pharmacies and reducing opioid misuse and abuse in racial and ethnic minority and rural communities. I'm representing my team today, which is comprised of researchers from our School of Pharmacy as well as our School of Medicine. Our research focus really focuses on patient-provider communication, expanding healthcare accessibility in rural communities, and advancing clinical practice in community pharmacy settings. Amongst our team, we all have expertise really spanning across implementation science, medication access, pain management, disparities in healthcare, gerontology, and the impact of social determinants of health. And so before we begin, I just like to always start with this slide. I know the previous presenter did talk a little bit about opioid use disorder, but $1.02 trillion, opioid use has tragically escalated to epidemic levels within the United States, reaching a staggering $1.02 trillion in 2017 alone. And this alarming reality really prompted the declaration of a national public health emergency. For those of you, again, it was already talked about, but opioid use disorder is characterized by the chronic consumption of opioids, leading to significant distress or impairment, and it manifests in various forms such as dependence, abuse, illicit distribution of opioids, and devastating consequences like overdose and profound disruptions to physical and or psychosocial well-being. And so in the efforts to sort of mitigate the effects that we are seeing related to opioid overdose, public health organizations like SAMHSA, CDC, and the FDA have implemented various strategies. These strategies really hover around 6 key initiatives: community education and awareness campaigns, widespread access to naloxone, prescription drug monitoring programs, increased access to medication-assisted treatment, safer opioid prescribing guidelines, and expanded access to syringe exchange programs and other harm reduction initiatives. Despite these critical initiatives, though, disparities exist, particularly for our marginalized communities. So what we are seeing is limited access to overdose prevention and treatment services. This is especially evident in underserved populations. So the impact of rural and minority communities, what we see is stark, and disproportionately higher rates of deaths are observed in racial and ethnic minority communities, alongside elevated drug-related mortality rates in rural areas compared to their urban counterparts. And so these gaps require further investigation, suggesting a need to bring adequate opioid use disorder care to these communities. And so this leads to looking at community pharmacies. This has become an increasingly prevalent space for pharmacies and pharmacists to expand their services and provide continued care for these patients. Many of you might not be aware of what community pharmacies can do, but pharmacies have a multifaceted role in addressing the opioid crisis. This can encompass prevention, treatment, and recovery support, and their accessibility in the community and pharmacists' expertise in medications really make them a valuable ally in addressing this. And so whether it's, dispensing naloxone, which is the life-saving medication for someone experiencing an opioid overdose, or even all the way to continuing education. So pharmacists are able to go out into the communities that they're served and help educate people about opioid overdose and ways to prevent this. And so just to summarize where we are now, we know harm reduction strategies work to prevent opioid overdose, but it still seems that opioid overdose and deaths disproportionately impact rural communities and communities of color. We also see that pharmacists are positioned in an important role to employ opioid use disorder services, but potentially seeing that rural communities and communities of color may differ from their counterparts within the scope of access of these services. And that leads to our need to determine what differences, if any, exist regarding opioid use disorder strategies within the community pharmacy. And so, given these disparities and gaps that are identified, our research seeks to address these issues directly. So overall, our project is seeking to increase our understanding of how race, ethnicity, and rurality influence the delivery of opioid overdose services within the community pharmacy. And so to do this, we plan to investigate barriers and opportunities for opioid overdose services in these pharmacies, understand the delivery of these services in racial and ethnic minority and rural communities, and elicit the perspectives of underserved populations to inform targeted interventions. And so our project aims to, one, create and distribute a quantitative survey to community pharmacies using a knowledge, attitude, and practices survey model to gain, again, a comprehensive understanding of OUD and overdose services, and aim two, to use an exploratory qualitative approach through semi-structured interview guides to comprehensively understand the experiences of pharmacists, patients, and community members with their community pharmacy-based OUD services. And so taking just a closer look at our aim one, again, pharmacies will be, pharmacy staff members will be surveyed via a web-based quantitative survey. Again, we're going to reveal their knowledge, attitudes, and practices. The reason why this particular model was chosen is because it provides a holistic view of how people feel or think about a topic and how they translate this knowledge and attitude into actions or behaviors. And so this comprehensive approach will allow us to identify gaps in understanding and areas where attitudes may influence practice. Again, our participants are the pharmacists and their support staff. Again, thinking that pharmacy technicians do have a great role to play in the delivery of these services. Again, by acquiring knowledge, attitudes, and practices in these areas, we can effectively support services in the community pharmacy setting. And so just to pull out a little bit about some of the topics that we do have on our CAP survey that we're delivering, we are asking really a wide range of different topics from pharmacology all the way to policy and regulation. So when it comes to policy and regulation, we are asking these pharmacies, how do they stay informed about relevant policies, regulations, and legislative changes related to prescription drug monitoring or opioid stewardship and other initiatives such as like de-prescribing? And all of these primary outcomes within the survey will be evaluated using descriptive statistics. We're also trying to do some comparative statistics to see if we're able to assess these endpoints in comparison with their counterparts. And so looking specifically at how we plan to carry out AIM-2, again, this is our exploratory qualitative approach in which we try to understand the experiences of the people within this community. And so we will be delivering a semi-structured interview guide that is focused on better understanding their experiences. We are using a framework to help develop these interview questions. The Consolidated Framework for Implementation Science will be used, again, focusing on access, delivery strategies, and regulatory aspects. And at the end of these interviews, we intend to do an inductive or open coding approach to reflect on the participant perspectives. We will also use cluster analysis to focus on the data that generates novel insights regarding the likelihood that individual demographics or health-related factors play a role in community pharmacy-based opioid overdose services. So we are actually finishing up all of the documents for the survey in the interview guide. But some things that we kind of talked about as far as like potential barriers and limitations and moving forward with the project were access barriers. So despite the proposed interventions, access to community pharmacies may still be limited for certain populations, therefore limiting the generalizability of our findings to certain rural populations and communities of color. Really to mitigate this, though, we are working with a practice-based research network that has 7 southern states, specifically in rural counties, and the community pharmacies have been partnering with this network for a long time to kind of carry out research studies such as this. So we don't really see too many issues with barriers, but when it comes to cultural sensitivity, again, we've learned that in past projects, opioids can be sort of a difficult topic for people to talk about. And so we are making sure that we are tailoring all of our documents to be culturally relevant. One of the things that we have done with one of our interviews and interacting with patients is instead of saying opioid overdose, because that could elicit some type of response from the participants, maybe calling it an opioid breathing emergency. This same can be said as it relates to stigma. And then in the data collection and analysis phase, again, collecting data and on opioid misuse and abuse in these communities may be challenging due to the limited healthcare infrastructure and resources for surveillance. So we're making sure that we are ensuring data quality and reliability for accurately assessing the impact of the outcomes that we found. And just to close, what we expect is that the outcomes of this project will address an unmet need in OUD research, specifically in the community pharmacy setting, to understand prevention, treatment, and recovery supports for racial and ethnic minority and rural communities. And we're hoping that this leads to enhanced understanding of the barriers and facilitators to these services, and the long term leading to development of tailored interventions for these vulnerable populations and contribution to regulatory science priorities and public health preparedness. I do have questions, but we'll wait until the next reviewer. So thank you.
Great, thanks, Tamara. It's going to be an exciting project. We'll see what we find. Our third presentation in this session is from Tucker Diamond Ames at the Mayo Clinic. He's going to be speaking about utilizing a web-based harm reduction approach to reduce disparities in harm reduction education and service utilization.
Awesome. I'm really glad to be here, and I'm really excited to be going right after Tamara because it's a similar project, but through a different approach. I'm eager to talk to her afterwards. So let me pull these up real quick. I gather we're all good here. Excellent. So my name is Tucker Diamond Ames. I'm a first-year medical student here at the Mayo Clinic, and I'm working on a community-based project with Dr. Lai that is utilizing a web-based harm reduction approach to see if we can reach marginalized populations within the Rochester area. So to better understand the project, I like to address the need for it, which we can understand through the opioid epidemic. Many people are very familiar with this graph at this point, so I'm going to run through it very quickly. We had the first wave of the opioid epidemic beginning in the 90s with the rise in prescription opioid overdose deaths, which had a worsening in 2010 due to a rise in heroin overdose deaths. A third wave occurred in 2013, where fentanyl and other synthetic opioid overdoses rose due to greater availability. And finally, a fourth wave, which happened a year after the COVID lockdowns began, where psychomotor stimulants and opioids have been used concurrently or have been contaminated with the opioids, leading to greater rates of overdose death. There are health disparities that exist between groups that are suffering these fatal overdoses. This is a fantastic figure pulled from an analysis over the year of 2019 from 25 different states that highlights rates of opioid overdose based off of income inequality within a county, represented by the 3 bars, and then also separated by ethnicity. Some things that really stand out are that in areas with the highest income inequality, Black and Hispanic populations tend to suffer a higher overdose death rates compared to when they're in lower areas with lower income stratification. And then Black and Alaska or American Indian Alaska Native populations suffer high overdose death rates overall. One way that overdoses have been reduced and been shown to be reduced in the past are through harm reduction techniques. These include methods like naloxone training programs and needle exchange programs. It can be considered as very direct acting, such as naloxone directly reversing an opioid overdose when administered by a bystander, or a needle exchange program, which has more of a chronic or long-term reduction in opioid overdose death and mortality by limiting disease. However, there are disparities in access to these services. One study from a group of outpatient treatment centers found that their participants had great disparities within access to these services when they were seeking them. And it's 10 times harder for minority groups to access naloxone and 8 times harder for them to access sterile needles. That's a huge issue, which is where electronic harm reduction comes in. E-harm reduction is a fantastic way to help level the playing field in terms of accessibility, as the web tends to be very accessible for many different people, and it's easier to get out there. These methods have been shown to demonstrably increase overdose risk awareness and also improve opioid knowledge. A really good example of this is Never Use Alone, which is a fantastic nonprofit organization that I am so sorry, Mr. Torres. I think you're unmuted. Thank you. I'm sorry. Never Use Alone is a fantastic online resource that is a remote drug monitoring program that has helped, I believe, on the order of nearly 12,000 individuals while they decide to use substances, and reversed over 100 overdoses due to the remote drug monitoring program. While this sounds fantastic, few studies have assessed whether websites and apps are efficacious in reaching those marginalized populations when put in the community in the form of advertisements or resources. This is where our project comes in. We created a Wix website with up-to-date harm reduction information, including harm reduction education, specifically focused on opioid overdose reversal, but there are other things in there regarding safe drug disposal and addiction treatment, specifically with resources and programs localized to the Rochester area, and then embedded local resources and direct links to those local resources in each of the pages to allow all of our visitors to possibly seek treatment or further education if they use the website. The reason we use Wix is it collects data that we're using to determine some of the outcomes of the efficacy of the website, such as the time spent on the website, how many people are viewing the website each day, and what pages tend to garner the most attention from our population. We also created an anonymous survey with a bunch of demographical data that we're interested in gathering related to participant ethnicity, income level, age, and housing status to help better understand if we are reaching marginalized populations or what population is utilizing this resource the most. And then we created stickers and business cards to display at pharmacies. The reason we chose community pharmacies is this is a place that individuals are receiving prescriptions for, and they may be interested in having more education about the prescription that they're being provided. All the materials were then translated into Spanish and Somali, and all of the site went live on March 29th. We're currently adding pharmacies in a cascading fashion to increase our reach and hopefully get a better survey response rate. Now, this project has only been off the ground for about a month, so I have very limited preliminary data, especially regarding surveys, has not been filled out yet. However, I can talk about some of what has been shown on Wix Analytics. Thus far, we found that the local resources page is the one that has time spent on the most, which shows us that people are interested in looking at the resources available to them in the Rochester area, and that many people are viewing the homepage, which is the page that people are directly put to. However, people are also going on to the addiction treatment page, which is actually the page that is viewed most often following viewing of the homepage. This gives us some really valuable data on what resources are most useful for individuals who are receiving this kind of information at the pharmacy counter, and it can help us target interventions down the line. Now, there are several potential impacts for this study, depending on how the data turns out at the end. The results can tell us where we need to go in terms of targeting interventions based off of the impact with the population. If we find that people are utilizing the website and they are from different ethnic backgrounds and they tend to view certain pages more, that helps us to identify ways that we can have more robust impact within the community. It's also a really low-cost method of providing harm reduction resources and increasing engagement. The entire website and resources took less than $300 to produce and put up at several locations. So it's a really low-cost way to hopefully reduce opioid or substance use overdoses. Finally, there's a potential for a broader community impact. These kinds of resources are much more readily shareable than things like naloxone, which have supply limitations. And this allows people to disperse information, which I believe is incredibly powerful to helping combat the opioid overdose epidemic. We do recognize there are some limitations to our study, though. It is subjective to selection bias, as individuals filling out the survey may not be representative of the individuals that are using the website or are interested in filling out the survey or interested in viewing the website in the first place. And then there's social desirability bias. Even though the surveys are completely anonymous, individuals filling out the survey may want to present themselves in a more positive light or underestimate the effects that opioids may be having on their life. There are also some challenges with generalizability. Rochester's population does not necessarily reflect that of the US as a whole. And finally, there's some data collection limitations. We're using an anonymous survey to prevent or protect the identity of individuals and hopefully increase response rates and honest response rates, which is what we're interested in. But as a result, we can't guarantee people are going to be filling out the survey. Depending on the results of our study, we have multiple different directions that we can go. One thing we are interested in answering even right now is why are people viewing the website but not filling out the survey? We've been having an average of 1 to 2 hits per day, and that's I think that's fantastic. However, I'm really interested in finding out why people aren't interested in filling out the survey, if they're worried about their anonymity or something along that lines. We're also interested in assessing if this form of very passive community engagement through advertised resources on pharmacy counter may differ from more active engagement, which is one reason I'm really interested in seeing how Tamara's project turns out. And then we are also interested in seeing if other e-harm reduction methods may be more effective, such as educational modules being incorporated into the website and advertised, or a social media type campaign, which has been shown to be a much more active type of engagement for younger populations and garner much more spread in the general population than a website. With that, I'm done with my presentation, and I believe we have time for questions. So thank you all for listening.
Yeah, thanks, Tucker. Just a flag, I know we're running a bit behind schedule, but started with the sort of keynote address, and we have to really try to keep on time with everybody going forward. But we do have a little bit of time for questions for our 3 presenters. Does anyone have a question?
I popped a few in the
chat.
Oh, Sten, thank you. Let's start with Dr. Liu. Can you comment on statistical challenges and bias with loss to follow-up when you're working with these real-world data sources?
Yes, yeah, this is a very good question. And I think, yeah, the loss to follow-up, especially differential loss to follow-up, may be a bigger problem in real-world data. So I think one good approach is, we need to address this kind of selection bias or informed differential loss to follow-up. And especially in the real world data, we have a lot of, patient characteristics that may be, that can be predicted the loss to follow-up. So we can use some, for example, the inverse probability scissoring weights, to adjust for differential loss to follow-up. And yeah, make our effect estimate more valid, for the true effect in the study population or covered population. Thank you.
Thank you. Dr. Hughes, Dr. Vermund asked, can you cite overseas models of pharmacy-delivered OST and how they've helped you? And I'm also curious if you could comment on, I'm just interested, how are you identifying the pharmacists to respond by email, which is just a very difficult population to identify emails from in order to do a survey, and your concerns about response bias?
Yeah. So with the first one, as it relates to overseas models, honestly, I have not taken a look at those specifically. And the reason for that, this was very intentional, only because we're dealing with a very specific population within the US. And if anyone knows the history of the US, the way our cities and demographics are made up is very unique when it comes to racial and ethnic minoritized groups, as well as rural communities. And so it could be some things overseas as it relates to opioid substituted treatment that could potentially help here in the US. But we're kind of starting from scratch again, just understanding that the United States has a very different makeup, and it's very important for us to think about cultural competency as it relates to the development of some of the things that will come as a result of this project. To answer the question about the concern for pharmacies and response rate, so it's a little easier, I think, for me. I am at a school of pharmacy. Also, I am a pharmacist. And so on a lot of my work, we really average pretty well when it comes to reaching out to our pharmacies and requesting information from this. And I think it could just be, again, from the personal relationship. I understand the landscape, and I kind of just understand that, I have to make sure that my research fits within to their time, which can be very busy and daunting. And so we've just kind of developed a lot of different things, particularly incentives to help urge the pharmacies to kind of complete in a lot of our research projects. So if you're interested in getting in touch with pharmacies, definitely reach out to me. I can give you some strategies on how to solicit them better to get a high response rate.
That's great. That's great. And I'm really looking forward to seeing what you find. Last question for Dr. Diamond Ames. So this is sort of combined, if you're looking in the chat between Dr. Vermund and Hashir Abdi, both asked about the populations in the Rochester area, including high numbers of Latin American Somali immigrants, along with local and rural communities, and how to think about language and translation and those issues. If you can open up the chat and take a look and perhaps address both.
I did take a look at the chat. That's an excellent question. It's something that we had to heavily consider. One thing that I used, we did the initial preparation of the content, and then I used a large language model to help me reduce the reading level from an objective way. And then I actually contacted some individuals who had no experience with the subject and asked them to review the content. And thus we simplified the content further. The goal was really to make it as simple as possible. And then when we gave the materials to our translators, we asked them to try to keep the reading level to the most simple degree possible. And they said they were able to do that. And I had people who were native speakers review those materials, and they said they were digestible at a lower reading level. For the second question, I think it's more of a suggestion. I think it's an absolutely excellent one. I think, honestly, that may be an approach I take following the termination of the study is I am interested in keeping the website up and continuing to collect data and seeing the best way of dispersing this. But right now, for my study design, I am interested in seeing if this kind of advertisement in a pharmacy setting is effective. I'm then going to utilize the website as an ongoing project to assess if community endorsement in that fashion is an effective way of increasing engagement with those populations as well.
Great. Thank you to all three of our first session presenters. I'm going to hand the baton over to Molly to lead us into session 2.
Great, thank you, Joe, and really fantastic presentations. I really enjoyed them. And we've got another panel of 3 really interesting ones. So we're going to start with Manam Shahzad from Harvard on the Orphan Drug Act.
Perfect. You can see my screen, hopefully. All good? All right. Perfect. Thank you for having me. I'm Manam. This is all work that is joint with one of my mentors at the Harvard-MIT Center for Regulatory Science. I'll be focusing on the Orphan Drug Act. I'm trained as an economist, so this will be all about incentives, which is sort of what we think about. So the Orphan Drug Act is an important piece of legislation which intended to sort of create incentives for the development of drugs for rare diseases. At that time, the thought was that rare diseases, or given that they impact so few people, they wouldn't be very profitable for pharmaceutical firms to invest in, and hence we would have no development for these drugs. This law was sort of enacted in 1983, so last year it sort of turned 40, which is a long time for a piece of legislation to be in effect. And one of the things that's been sort of true is that we definitely have seen a lot of indications that are benefiting from this. So the graph on my first slide that you can see here, the blue line is the number of designated indications. Designations happen fairly early on in a drug's life cycle. So once a pharmaceutical firm has sort of a product where it has some indication that the product would be useful for an orphan disease, they can get it designated by the FDA. The red line is the one that you'd probably sort of think about as the indications that you're also seeing in the market as having been approved and now available to patients. And so one of the things that's sort of been the way that the Orphan Drug Act essentially, incentivizes this kind of development was through a lot of push incentives. So there were a bunch of tax credits and grants that were meant to offset research activity. There's exemption from user fees. So the FDA doesn't take user fees for orphan designations. And at that time, the sort of big piece was this idea of exclusivity. So exclusivity essentially would protect the product from competition. However, at that time, this exclusivity was essentially a indication product specific exclusivity. So essentially you couldn't market the product for that particular indication for 7 years after the indication was approved. And this was at that time thought to be sort of one of the major incentives in place. The other things that have sort of happened with the Orphan Drug Act over time, which we've seen in some of the literature, is this idea of regulatory flexibility. And we talked about this sort of in the keynote speeches that there's an idea in which sort of the FDA is thinking about sort of some regulatory laxity when you need drugs for these diseases. And so we've sort of seen that the FDA has had lower lower bar for evidence for orphan drugs. And then further down, there's sort of other benefits, which is that we know that orphan drugs are charging high prices. Recently with the IRA, there's an exemption from Medicare negotiation if you're only approved for one orphan indication. This has also at that same time sort of led to huge concerns about the Orphan Drug Act. So high prices, we think about the fact that we're spending a lot of money on these drugs. There's concerns that pharma companies might be sort of salami slicing, which is the idea of sort of arbitrarily slicing down a disease so that it then qualifies for the 200,000 people threshold. And then also the idea that maybe the regulatory flexibility has gone too far. And so in this backdrop, sort of what motivated the project is that when all the incentives that I talked about, at least the earlier ones, were brought into, were legislated, and the concerns are sort of happening more so now, truly the landscape of drug development has changed a whole lot since 1983. So one of the things that you can see on my slide here is that the FDA now has many other ways of expediting or incentivizing drug development. So FDA has 4 expedited pathways: fast track, priority review, accelerated approval, breakthrough designation, which can also sort of be used in conjunction with the orphan designation. And sort of looking at this graph, which focuses on indications approved between 2011 and 2022, you can see sort of that orphan alone, which is the blue bar, is a really small part of the puzzle. In fact, orphan, at least for first indications, which we sort of think of as when you come into the market, they're often used in conjunction with other programs. The other things that have happened over time is we sort of see more precision medicine. We've seen a lot of growth in biologics, which actually I'll discuss interacts with some of the incentives of the ODA. And overall, they're sort of increasing insurance coverage, which may have changed the incentives. So as we think about, what does the ODA really buy you? So the exercise that we ended up are still in the process of undertaking, but nonetheless, I'll sort of share the high level stuff, is we looked at each of the incentives associated with the ODA and tried to think about whether we could learn something about whether this is still a key incentive in promoting rare disease development. So one of the benefits of the ODA, as I mentioned earlier, is a tax credit. In 2017, this tax credit was actually decreased in value from 50 to 25%. So you could now recoup 25% of your clinical trial costs. And if you look at the graph, it almost at first pass, it seemed like, the blue line's going up. If you look at 2017, maybe we could make the argument that things are coming down. But one of the things that's sort of been hard in associating, in assigning causality to this is that there were a bunch of other things happening at the FDA also. We've done some work looking at sort of clinical trials, and we don't pick up a lot of disincentive coming from this reduction in tax credit. So some suggestive evidence that maybe this tax credit is not pulling everything. Then we move on to sort of the exemption from user fees. This is actually sort of like hefty in the way that the user fees are now about 4 million, and we think this might be attractive for smaller firms. But if you look at the firms that are applying for the orphan designation, actually a significant portion of these firms are large firms where we think that this 4 million might not be a big player in sort of their decision. Moving on, then we think about sort of the indication-specific market exclusivity. There's an analysis that is published till 2014 that essentially showed that the 7 years of indication-specific market exclusivity is increasingly not the binding exclusivity. So by virtue of either other exclusivities or patent protection, drug molecules end up being protected for much longer than what is offered by the orphan drug exclusivity. And increasingly, as we see sort of biologics, where we know that the patent life is so much longer, we expect this to be an even smaller part of the sort of benefits associated with this. So that sort of led us to thinking then sort of what's left on the table. And in sort of talking to pharma firms, there's a sense of, it's easy. So I think the sentiment that you get is that it's sort of easier to get an orphan indication on the market as far as the interactions with the FDA go. So we think of that as sort of regulatory flexibility. Just to note that this is not coded in the statute. And there is some prior evidence that sort of looked at it, but it's sort of much older. So we've undertaken sort of a update to that, and I'll only show you sort of 1-year results, which is that we looked at all the new molecules approved in 2022. We spent a lot of time looking at the summary documents associated with them to try to understand what was the level of evidence when these indications were approved. And we come up with a sample of 37 drugs, 20 of which are orphan designated. This is to highlight that orphan designations are an important part of the drug development paradigm now. 16 of these are sort of biologics, 30% are cancer associated, and about 41% are approved for indications where the summary documents indicate that there is no therapeutic alternative. The key thing to note is that orphan indications are taking less time. So it's about 6.6 years versus 7.2 for non-orphan indications. You could argue that this is all confounded in many ways, and I will argue that that is true. This is also not significant in the small sample, but as we're doing more data collection, the timing will probably be significantly different. The thing that I want to bring your attention to is this idea of expedited pathways. So you'll notice that orphan indications are much more likely to use these other expedited pathways, so accelerated approval pathway or the breakthrough designation, which you can sort of see here. If you then look at the characteristics of the pivotal trials, we also see that orphan indications are sort of, the bar is somewhat lower. So 80% of them are using single trials versus sort of the FDA standard of two trials. They're less likely to have confirmatory evidence. They're sort of smaller. Part of that you would expect because orphan indications tend to affect less people, but also in things like randomization, they're also less likely to use randomized trials. So even in the sort of 1-year study, which we sort of do find that the bar for evidence is somewhat lower. So sort of to bring it all together, the key sort of insight so far from the analysis is that the benefits of the orphan designation definitely seem to be broader than what is encoded in the statute. The things sort of, we focused on regulatory flexibility. There's a lot of literature now on the IRA and the incentives in the IRA. We've always known that the orphan drugs are sort of priced high. These are not things that were sort of encoded in the Orphan Drug Act, but that's sort of how the drug pipeline has evolved, and are likely the big benefits that are seen by pharma firms. The other thing that this analysis highlights is that it's very hard to study just the ODA, because oftentimes now with so many options available to pharmaceutical firms, lots of pathways and things are used together. So it's very hard to sort of distill that this impact is coming from the ODA. And this is a little bit of like a cautionary note to research that sort of only focuses on the orphan, non-orphan binary, because orphan drugs are also coming in with these other expedited pathways. So
Fantastic.
I believe that.
Yeah, thank you so much. And we'll hold questions till the end. That was terrific. Thank you.
Thank you.
Next up, Alyssa Chen from Yale is going to present a systematic review on anti-obesity meds for older adults.
Hi, thank you. I'm just going to start my slides. The button's going to work. Okay.
Perfect.
Okay, great. Hi, so my name's Alyssa. This is my presentation, Weight Loss Effect of Anti-Obesity Medications for Older Adults, a Systematic Review. And I'm going to run through the background, methods, and results and conclusion of my study. So 42% of older adults have obesity, although there's not clear treatments for obesity in older adults. Although many new medications have been developed for the treatment of obesity, like Zepbound and Wegovy, it's not totally clear how these perform in older adults because the clinical trials that study these medications under-enrolled older people. And studies that focus on older adults are really necessary for these medications because obesity is very different in older adults as compared to younger adults for a few reasons. Older adults are more likely to have secondary conditions from their obesity, like cardiovascular disease or hypertension. They're also more likely to be on medications to treat those conditions and other conditions that might interfere with anti-obesity medications. And many older adults with obesity have what's called sarcopenic obesity, which is a syndrome of high fat mass but low muscle mass. And all anti-obesity medications induce some muscle mass loss, so it's unclear how that's going to affect the function of older adults. So the objective of my study was to perform a systematic review of the literature to determine the weight loss effect of anti-obesity medications in older adults. For the systematic review, I worked with a librarian who developed search terms focused on older adults, FDA-approved anti-obesity medications, obesity and overweight. She searched many databases and didn't put a time limit on the search. And we also searched citations of included studies. And my co-author and I applied the inclusion criteria to the publications that were returned in the search. And that was study type, could be clinical trials or observational studies. The population were people who could meet criteria for treatment with these medications, which includes people with a BMI of 30 or above, or a BMI of 27 to 30 with one weight-related comorbidity. The studies had to have measured weight loss and reported results for those who are 65 and older. And that could mean that the study only focused on older adults or stratified their results by age. And then the treatment was FDA-approved pharmacotherapies for weight loss. And this is just the list of pharmacotherapy for weight loss. And there's some things that I'm sure you'll recognize, like Wegovy, Saxenda, and Zepbound. But there are a lot of older agents as well, like phentermine and orlistat, that are quite commonly used. And I'm going to show the results that we have so far from the systematic review. So we started with over 5,000 studies. And after applying the inclusion criteria through title and abstract screening and full text review, we got it down to 8 studies that were included. I searched citations from those included studies and found no new studies to include. And of those 8 publications, it was 4 full manuscripts and 4 abstracts. And it gets a little confusing because several of the publications describe data from multiple clinical trials. So I'm also going to represent this as the number of studies described. So of all those publications, there were 2 observational studies described and 6 clinical trials. And of those 6 clinical trials, there are 5 secondary analyses of RCTs and 1 single arm study. This graph shows the medications that were represented by those studies. On the x-axis, you can see the number of studies, and the y-axis, you can see the medications. And the medication that was most represented was liraglutide, with 4 of the studies reporting weight loss results for that, followed by phentermine, topiramate, orlistat, and semaglutide. And I wanted to share the details of the clinical trials. So there were 2 semaglutide clinical trials, one with scale diabetes, the other scale obesity and prediabetes. And these are obesity trials, so they were meant and powered for looking for differences in weight loss. These are represented, unfortunately, just by an abstract. And there were 2 liraglutide studies, 2 cysteine studies from Japan. These were diabetes studies, so really powered to look at changes in hemoglobin A1C, but as a secondary outcome looked at weight loss. There was 1 phentermine topiramate study, which was an obesity study, and that 1 single arm study with orlistat. And I'm going to show the weight loss results for the randomized controlled trials in this table. So for the semaglutide studies, you can see the sample size is 201 and 119, and that's of older adults in the studies. And there was a statistically significant difference in weight loss between liraglutide and placebo. You can see in the first study, the average weight, or the mean weight loss, was 8.4% compared to the placebo of 4.2%. And here is where it gets a little bit complicated to compare these studies. So you can measure weight loss in a lot of ways. The semaglutide study measured it in percent body weight loss. These liraglutide studies measured it in absolute weight loss. But you can imagine that if losing 4 kilograms of weight means something different for somebody with a BMI of 40 versus a BMI of 27. But regardless, in these liraglutide studies, they found a statistically significant difference in the weight loss that people achieve from liraglutide versus anti-diabetes medications. And then in the phentermine topiramate study, there were 219 older adults, and there was statistically significant weight loss seen from phentermine topiramate to placebo, so 9.4% weight loss for the highest dose of phentermine topiramate. And there were 2 observational studies that were pretty small and combined weight loss effects of multiple medications in 1 effect size. So in this first study, they used electronic health record data from a metabolic clinic in Venice, Florida, for 29 people, and there was a weight change of 11.4%. But the interesting thing about this analysis is they took out anybody who was a non-responder. So anybody who didn't show weight loss, they took out of the effect size. So kind of biases their estimate. And in the second study, they used electronic health record data from Colombia, and the sample was 38 older adults. And they looked at this analysis using a logistic regression. So those over 65 had a higher odds of achieving 5% weight loss than others. So to just summarize what we found, largely the literature is really lacking in data regarding the weight loss effect of these medications for older adults. So there were no randomized controlled trials that were focused on older adults. And there were only small observational studies of older adults. And those studies combined the effect of multiple medications. And these medications work very differently. The weight loss effect is very different. And it doesn't really make a lot of sense to combine multiple medications into 1 effect size. Regardless, the secondary data from randomized controlled trials do suggest weight loss. And the implications from the study, I think the biggest one is more data are really needed. I think randomized controlled trials that enroll a reasonable amount of older adults or focus on older adults. And also, I think there is a huge space for observational data here. I think there are older people who are taking these medications, and we can use the data from EHR or other large sources to see what the weight loss and safety effects are. And given the lack of data, like the coverage considerations are quite complicated. Medicare right now only covers these medications for cardiovascular disease and diabetes. But it's hard to say that they should cover it for weight loss when there's not a whole lot of data to support their use. And finally, weight loss doesn't necessarily imply clinical benefit, especially for older people. So someone who's older might be taking one of these medications to lose weight and improve their pain, but we don't know if that's something that will actually improve their pain when there's not studies to look at that. Thank you to my programs and my co-authors. Happy to take any questions at the end of the session.
That was terrific. Thank you so much. And our third presentation in this panel is Vimig Socrates presenting on automated deprescribing recommendations.
Hi, everyone. Sorry about that. Yeah, I'm hoping everyone can see the slides there. Thanks so much. Yeah, so my name is Vimig Socrates. I'm a graduate student in computational biology and bioinformatics, as well as the new department in biomedical informatics and data science at Yale School of Medicine. So I'll be talking today about a pilot study that we ran to try and automate deprescribing recommendations using large language models in the emergency department where I'm based. So we don't have any conflicts, but we did use some AI, as you'll see momentarily. A little bit of background. So around 1/3 of older adults, defined here as 65 and older, have polypharmacy, which is greater than 4 medications, and that's been associated with a number of negative health outcomes. Those outcomes include things like falls or adverse drug events or returning to the ED, et cetera. And so a number of prior efforts have been have tried to implement automated systems to try and identify potentially inappropriate medications or PIMS, but they've been limited largely due to the sort of complexity, and that's kind of where we were hoping that we can use large language models to, subsume some of that complexity. So a few different criteria currently exist out there, so I'll go through them here. So the first one is called the STOP criteria, which is a European consensus study-based criteria. It has around 114 medications on their recommendation list, and they're all meant to identify potential PIMS. Similarly, the American Geriatric Association also released one called the Beers criteria that consists of an additional 39 recommendations, and then this one also includes some strength of evidence associated with their recommendations. And finally, more recently, actually out of Yale, the GEMS group developed this additional list, which is sort of a subset of the Beers criteria list. That's called GEMS RX, and that includes 8 sort of recommendations specific to the ED. And so the medication classes are a subset of the Beers criteria, but their indications and sort of additional qualifying information is slightly different. And so that's why we included that one as well. And all these recommendations identify PIMS that require a range of screening activity that includes identification of prior diagnoses, indications, lab results, and other nuanced clinical data. And that's sort of where we're hoping to use large language models. So the goal of the study is to assess the potential role for large language models, in this case, particularly the Azure GPT-4 model, in performing automated review of potential PIMS and STOP criteria that might apply to them using EHR data. So this is a very initial pilot study, and so we initially only chose 10 patients that are all adults with polypharmacy in 2022 that showed up in an ED for an ED encounter at the Yale New Haven Health Hospital System. We only included structured data as of right now, so those are things like problem lists, lab values, vitals, et cetera. So we're currently excluding clinical notes, although that's something we're planning on including in the future. And finally, we did a gold standard review by emergency medicine physicians to determine whether or not the STOP criteria that the large language model determined are in fact actually applicable to that patient. So a little bit more detail on the background there. So like I mentioned, we went through this inclusion criteria process, sampling 10 patients. Here are all of the structured information that we included. So that's the main key one here is outpatient medications. And part of the reason that's particularly important is a lot of these deprescribing criteria include things like multiple drugs should be prescribed at the same time or comorbidities and things like that. And then we also included a long list of structured information that was all sort of curated to make it look sort of like a pseudo clinician note. That includes things like demographics, labs, vitals, past medical history, and procedures, etc. So we broke this process down into sort of two stages. And the first stage here is sort of filtering the existing stop criteria. And so this initial pilot study, we just selected stop criteria. I'll touch on why in a little bit. So out of those 114 stop criteria, which of them are actually applicable to this patient? So the reason we did that is there's this issue in large language models that's recently found called lost in the middle, where if you have a really large context window or prompt, you lose some of the information in the middle. And so we wanted to filter that 114 list so that we don't apply it to every medication, but just the ones that are relevant. So this first step, in blue here, you can see the two inputs that we included. So we include the full list of 114 stop criteria, as well as a list of patient medications. And then we ask the LLM in red here to extract the criteria that might be relevant to that patient. And so that's excluding any sort of indications or other qualifying factors, just whether or not that medication is something that the patient is taking. And finally, we have it output the criteria, including a numbered list that it can use in the next step. And so what that might look like is the LLM might say, okay, based on this patient's med list, I think that these two are relevant. So a loop diuretic for treatment of hypertension with concurrent urinary incontinence, or benzos for agitated behavior or psychotic symptoms of dementia. So the next step in this pipeline would then be to say, okay, given this set of, in this case, two filtered stop criteria and the full set of patient information, all the structured information, the ask here is determine which of these patient medications may actually be deprescribed given the other sort of qualifying factors. So in this case, the loop diuretic needs to specifically be prescribed for hypertension, and the patient needs to also have urinary incontinence at the same time. And then we ask it to list all the medications that should be deprescribed, or to say that there's not information to make that designation. And finally, we evaluate this against physician gold standard, in which there's sort of four main outcomes that can happen. The LLM and the physician both say that this medication should be described as true positives. True negatives are the same thing, but they shouldn't be deprescribed. And the incorrect choices where, in this case, the LLM may believe that a medication shouldn't be deprescribed when in fact the physician believes that it should, as a false negative, and the inverse of that as a false positive. So some preliminary results just on the cohort level. So across these 10 patients, we had 120 approximately medications that were identified, around half of which that had relevant stop criteria at all. And then actually, so half of that, so 28 medications out of that initial 190 were potentially contraindicated, but required additional information. So in fact, a very small number of those were truly contraindicated, given the structured information that we were given across that 10 patient cohort. And so a little bit of results here. So when we ran the GPT-4 model on that set of 57, we do fairly well in determining that the GPT-4 model does fairly well in determining whether or not stop criteria are relevant to that patient. So 41 out of the 57 medications, the LM says, yes, this medication has relevant stop criteria that could further be evaluated. However, then in the application of that criteria in that second stage, we do much worse. And so the LLM in this case generally does well in identifying medications that should be continued, but does a lot worse in cases where it should be contraindicated or potentially contraindicated. And something interesting of note here is, so I've outlined here in red some of the potential missed cases that happen, and you can see there's sort of an overrepresentation to some degree. Again, this is a very small pilot study, so there's obviously concerns on generalizability, but there does seem to be some indication that the model predicts that a medication should definitely be contraindicated or definitely not be contraindicated, when in fact the physician thinks that there should be additional information. So that was something interesting to find. We can sort of summarized that by saying that the GPT-4 model is generally overconfident in its recommendations, predicting that 72% of PIMS were absolutely contraindicated or acceptable, when in fact additional information was required according to the physician gold standard. And on manual review, we find that many of those cases are an over-reliance due to structured information and the inability to assess sort of ambiguous criteria. And so some examples of that are, for example, the acetaminophen recommendation, where patients need to have poor nutritional status, which can be defined as a low BMI, but BMI might not be in the EHR, or it might be difficult for the LM to compute. Another example is statins for primary cardiovascular prevention, where the patient needs to be over 85, which we do have information about, but they should have established frailty with an expected life expectancy of less than 3 years. And that's difficult even for physicians to estimate to some degree. And so the LLM definitely struggled given those ambiguous criteria. So given that the sort of variability in the computability of deprescribing recommendations, we sort of decided to take a step back, and we said, okay, let's take a subset of the criteria and determine which ones can actually be computed based on EHR information that we have. So we're starting and we're conducting this current consensus study around with 9 clinicians that have a wide area of expertise, from IM to geriatrics to pharmacology, et cetera. In green here are the 3 physicians that have already finished, and so they're the ones that I'm going to present information about. But overall, we asked them for each of these recommendations. We asked them for these 9 different. We asked them about 9 different questions, some of which are clinically their clinical applicability, and other ones related to the EHR computability. And so what we see here is these are all of the questions that we asked them, but I'd like to highlight a few in particular. In red here, you can see, so in terms of the difficulty of assessing the applicability of that particular recommendation, or how much information the EHR does not have and needs to be gathered, we can see that there is no criteria, at least according to these 3 clinicians, that has all of the information in the EHR or is incredibly easy to assess the applicability of. So there's already some that we're going to have to remove and filter out. However, something else that we can note here is that when looking at how difficult the information needed to find the information that is necessary to in the EHR, or how much additional information is needs to be collected, we can see that in general, sort of the Beers criteria, and obviously a subset of that, the GEMS RX criteria, are a little bit easier. And so we're probably going to focus our efforts there. So some quick conclusions. Sorry, I know I'm almost at time, but some quick conclusions. So the GPT model in general was able to identify appropriate stop criteria, was not able to apply those. And part of that has to do with the ambiguity associated with these recommendations, which we're currently conducting that consensus study for, and we're planning on expanding out that cohort of analysis as well. So that's our group, and I'm happy to field questions at the end of the session. Thanks.
Fantastic. Thank you so much. So I will open it up to questions for any of the 3 presenters. You can put them in the chat or raise your hand. And otherwise, I am just going to start with a question for Vimig, which is, why use a large language model when you're looking at structured data? Wouldn't it be easier to just generate rules?
Yeah, that's a good question. I think part of the reason we went with that, with the large language model, is because I think the rules, there's some previous literature on trying to establish rules. And given the way that the criteria are, for example, those couple cases that I mentioned, in addition to a number of other cases, people have found it really difficult to identify structured information. So things like ICD codes or LOINC codes for lab testing, things like that. It's difficult to match those up with these criteria. So it's actually fairly difficult to define computable criteria for them, which is why we went with the LLM.
Thanks. Go ahead, Joe.
Thanks, and congratulations to all 3 of you on great presentations. My question is actually for Manu. I was curious, I was so intrigued, actually. I had not thought through this problem in the way you presented it, particularly the small firm, large firm, and regulatory flexibility. And I'm wondering, as you're starting to collect data over the years, if you're seeing a difference in the trial design and qualities between small firms and large firms. I sort of would have anticipated that large firm trends wouldn't change so much, but maybe you'll observe that.
That's an interesting question, and I can't answer it yet for this data because I haven't merged the applicant qualities to the trial things. But my dissertation was all on the accelerated approval pathway, and what we found was that I think in the larger firms, I think, tend to actually be better on a number of the regulatory characteristics, which is something that I actually did not expect because I think I went in with a theory of like regulatory capture, like maybe. But I think that was sort of true, and we sort of see better outcomes for larger firms, likely because they have a better infrastructure. And so I think that we might see that here also, because the one thing that I guess I could say is that the one thing that I have looked at is whether an orphan indication actually ends up in approval. And there actually small and large firms are doing similarly, which is surprising. But I still think that on trial characteristics, I think large firms might be better from prior stuff, but I can't say that for sure. But that's a great question, something I've been thinking about at least.
Other questions? If not, I have a question for Alyssa. So I was really interested in the results, and in particular, the fact that the new drugs that are getting all the attention, the GLP-1 receptor agonists, were not necessarily better than that older drug, phentermine topiramate. So is that specific to the older population, or is that just a drug that has a lot of other issues, or what's going on with that?
Sorry, I had to cough because I'm like recovering from an illness and I missed like half your question.
No. Okay, so my question was, I was surprised that the older drug, the phentermine topiramate, is at least as good as the drugs that are getting all the attention and that are so expensive. And is that specific to the older population, or what's going on with that?
No, it's not actually specific to the older population. If you look at the trials that were used to approve drugs, like phentermine topiramate, naltrexone, bupropion, they had similar weight loss efficacy in all comers. And that effect size you saw with older adults is pretty similar to the effect size with everybody in the trial. So I don't exactly know why they didn't gain in so much popularity. I think the newer drugs like can get you like to 25% weight loss. Now, everybody doesn't get that, but I think the way that they're marketed and talked about, like people can get there, and that is very exciting for people.
So it's kind of a heterogeneous treatment effect thing where there's some people who are like massive, and that's not the case with the older drugs.
Yeah, I would say with the older drugs, there are not people who were getting 25% weight loss for sure.
Fascinating. Thank you. Other questions? I see a question in the chat for Vimig. Did you use RAG or just stock GPT-4?
That's a good question. No, we just used GPT-4, although definitely something like RAG pulling in additional sort of information about these medications would be pretty interesting.
Other questions? Otherwise, I think we can gain back the time. And I'm sorry, I think we're going to take away the break. We're going to push through. It is the price we pay for having had a really great and interesting and inspiring first talk. So I will hand it back over to Joe for the next panel.
Great. And we have to give special kudos to Alyssa, who pulled herself out of her sickbed from her toddler-infected respiratory virus to present
Thank
today.
you.
So thanks, Alyssa. All right, so we'll move to session 3, where our first speaker is Alonso Torres from UCSF, who's going to talk about an immunoprotective multi-channel kidney bioreactor for implantable renal replacement. This is the portion of the CERSI Scholars meeting where I am way out of my league because I don't really remember basic science that was taught many, many years ago. But Alonso, take it away.
Thank you. Thank you for that introduction. And thank you everyone for being here. Again, my name is Alonso and I'm a 4th-year PhD student in Dr. Shubal Roy's lab at UCSF. And I'm really excited today to present some developments for the implantable bioartificial kidney that we are working on in our lab that is intended for end-stage renal disease patients. On the top right, we have a representative image or conceptual design of the implantable bioartificial kidney, or IBAC. And most of my work has been with one of the components of this device, which is the implantable kidney bioreactor, which is just below that figure. And I will go through in future slides what it entails. So to go through the background, chronic kidney disease is on the rise. More than 1 in 7 people in the United States have it. And as it progresses, there are really only 2 solutions for patients as they approach end-stage renal disease. The most common being dialysis, and hemodialysis is the most common subcategory of that, where ultimately blood with waste products comes into contact with a semi-permeable membrane, and the waste products are taken out by that membrane. This type of treatment requires, for very severe patients, requires 3 to 4 sessions a week, sometimes 3 to 4 hours a day. So it's very time consuming. It is a huge burden on the Medicare system to keep hemodialysis going. And it does affect the quality of life of the patient, unfortunately. As kidney disease progresses, though, the best solution is a kidney transplant, which on the right, we have a representative image of where the transplanted kidney would be attached between a main artery and a main vein. And then the urine that the kidney produces would then be directed towards the bladder. And kidney transplantation is the best solution for these patients. However, kidney donor organ shortage is a huge problem throughout the world. In the United States, about 100,000 patients remain waiting on the transplant list for a kidney donor organ. And so in the Roy lab, we have developed an alternative for these patients, which is the implantable bioartificial kidney, which is made from biomaterials, and we incorporate a cell-based component. On the left, we have another conceptual design where, just like the kidney transplant, the intended implantable bioartificial kidney, or IBAC, is connected between a main artery and a main vein. And there are 2 components within the device, one being the hemofilter, which is the filtration unit, and the other component being the cell-based component, which we call the bioreactor. The enabling technology of our device is called the silicon nanopore membrane. The silicon nanopore membrane it's made out of silicon, similar to the material that's used for electronics. However, this device does not use electronics. It mainly works on essentially the blood pressure difference between the artery and the vein. That blood pressure difference of about 2 psi allows for blood flow to continuously go through the device. In the lab, we do encapsulate the silicon nanopore membrane with hemocompatible coatings, and these hemocompatible coatings have a diameter size of about 2 nanometers. But in the figure on the right, we can see the distinction of the nanopores within the silicon nanopore membrane, and then in 3 and 4, we could see more distinct characteristics like pore width, pore length, and pore height. And these pores are synonymous and about the same size, which is about 7 nanometers in width, as the filtration unit in our kidneys, which is the glomerulus. And so in the last year, we were able to publish data on a single blood channel implantable kidney bioreactor. Again, this is one component of the IBAC. On the left, we have the representative image. And in the white squares that you see in the device, that's where we seed human renal epithelial cells. In figure 2, we have a diagram of how the blood flow goes through the single channel blood channel. And then the silicon nanopore membranes essentially create ultrafiltrate, or filter out they filter out whole blood. We do not use any anticoagulants. So the filtration then gets in contact with human renal epithelial cells, which are seated at the very top and very bottom of the diagram. After 7 days of implantation into a swine model, we were able to see greater than 90% cell viability for these human renal epithelial cells. And in addition, we were able to see upregulation of key transporters, which are aquaporin 1, which is a water transport protein, sodium hydrogen exchanger, which is also a transport protein. And then also the very last one is 1-alpha hydroxylase. And this is the main catalytic enzyme in the mitochondria of these cells, which activates vitamin D. And in the kidney is the primary place where vitamin D is activated for us in our body. And so we recently moved, and most of my work has been involved in enhancing the cell capacity of our devices to improve the therapeutic output. So we created a 4 blood channel implantable bioartificial kidney bioreactor. And the image on the left is the image of the device. In figure 1, we had to create a stack, which you see in the black silicon material. We had to create a stack with the silicon nanopore membranes and PDMS gaskets or silicone gaskets. And we did this by a method called of plasma bonding, which is basically reactive oxygen that we use to permanently place the silicon nanopore membranes and gaskets together. This is a very tedious process, and just a little misalignment would allow for the stack not to be inserted in these polycarbonate housings, which you see in blue. These polycarbonate housings are machined, and then after incorporating the stack, we close off the device after incorporating cell inserts, which you see in figure 3. The cell inserts are made out of acrylic, and the tiny squares that you see, the 4 tiny squares that you see, is where we seed the human renal epithelial cells. These cells are seeded on the acrylic inserts in an incubator in our lab. And once we sterilize the device, we incorporate the inserts into the device and close the door off. In the figure on the top right, we could see the implantable bioreactor in its final form, and we have the diagram of how it's connected to the swine model. Again, blood flow through the device is perpetuated by a difference in the pressure between a main artery and a main vein. And we place the device in the retroperitoneal space of the pig. On the bottom right, we have an image of the device being attached to Teflon grafts that are attached to the vessels of the pig. And so after several months of preparation, we incorporated 3 of these devices into 3 distinct pigs. After 7 days, the human renal epithelial cells had an average viability comparable to cell inserts that were left in an incubator for the same amount of time in our lab. However, we did see an ammonia concentration that was similar in the pig serum and within the cell chamber of our device. A difference we did notice, though, is the activated vitamin D or calcitriol in our device was different than in the pig serum. And we are still trying to evaluate why. However, I do have a video of an angiogram that was taken at day 7 of one of our devices, which shows smooth contrast through the device, which validates that our device did not have thrombosis. And so the device remained patent after 7 days without having to use anticoagulation. And so you could see a smooth transition of ion contrast through the device and through the loops that are created. And so we did show a successful implantation of 3 leak-free multi-channel bioreactors with cells for 7 days. And again, we did not use immunosuppression or anticoagulation medications. We did confirm that the blood flow was patent through the device, so no thrombosis was observed. For ammonia concentrations in the pig serum and in the bioreactor chamber, we saw that they were similar and indistinguishable. However, we did see a difference in concentration of activated vitamin D or calcitriol in our device and in the pig serum. My hypothesis is that the carrier protein for this particular protein, calcitriol, might have been blocked by the silicon nanopore membranes within our device. And so we are looking further into how well these complexes get through the silicon nanopore membrane and get in contact with the cells that we are encapsulating in the implantable device. Future directions include improving the viability and function of our cells. For my dissertation, I'm actually doing a scaffold work to recapitulate the basement membrane that the cells are seated on and incorporating that material into our devices for future implementation. With that, we'd like to conduct longer temporal studies and also scale up to larger multi-channel devices to improve our therapeutic output. However, we did confirm a pathway for scaling up to those cell capacities. And so we hope to incorporate a similar protocol and similar steps to build larger devices in the future. And lastly, thank you all for your attention. I'd like to thank Dr. Roy in our lab, my family, a couple of my mentors, Dr. Paul Brakeman, and a couple of funding organizations, which are the John and Marcia Goldman Foundation, the Pat Tillman Foundation, and then everybody for organizing this great seminar, and the UCSF Stanford CERSI group that I interact with throughout my graduate program. And that's it. Thank you.
Terrific. Thank you, Alonso. We're going to, again, like before, hold questions to the end. Our second presentation in this session is from Francis Xu from the Mayo Clinic, who's going to be talking about CSF inflammatory molecules and Alzheimer's disease biomarkers for predicting cognitive impairment risk in the elderly.
So thank you all for having me today. My name is Francis Xu, and I am a PhD candidate at the Mayo Clinic. To start with some background, Alzheimer's disease is the 6th leading cause of death in the United States, where aging is the biggest risk for developing Alzheimer's disease. Typically, patients are usually diagnosed with cognitive impairment or with dementia in general, and you sometimes get guesses of which type of dementia one has. So common ones are usually Alzheimer's disease, Parkinson's, or even ALS. However, there's only one way to get a proper diagnosis, and that is pathologically post-mortem, which doesn't really help if you're trying to help people pre-mortem with their treatments. And even at the most renowned medical institutions, accuracy of post-mortem diagnosis is only up to 80% accuracy and much lower at other institutions. So my project specifically looks at Alzheimer's disease. But before I can start going into my data, I do need everyone to know a little bit more about this protein called apolipoprotein E, also known as ApoE. So ApoE physiologically acts as a lipid transporter and integral for the clearance of amyloid beta, one of these major pathological hallmarks for Alzheimer's disease. So ApoE comes in 3 major ice cream varieties: ApoE2, ApoE3, and ApoE4, where there's only one difference in each of these isoforms at positions 112 and 158 that affects its binding and physiological function. So ApoE4 is the strongest genetic risk factor for developing Alzheimer's disease, where 50 to 70% of all 80 patients carry at least one ApoE4 allele, where ApoE2 is overrepresented in our centenarian population and also known as a longevity gene known to be protective against Alzheimer's. And ApoE3 is kind of the most common genotype and allele most people in the population carry. So our cohort is taken from the Mayo Clinic study of aging. This is an ongoing longitudinal study here at the Mayo Clinic that started in 2004. So we had 3 major qualifications for the subjects included in the study. So 1, they had to be 65 or older, and they had to have a clinical dementia rating of 0 at their baseline visit, where we collected their cerebrospinal fluid, meaning that they were cognitively normal when this happened. 2, they must have had at least 2 clinical follow-up visits, so we know whether or not they ended up converting or not. 3, we had their ApoE genotype information and longitudinal CDR neuroimaging information on file. And to note, there are no major differences in collected clinical diagnostics within each of these ApoE groups. So to do this study, we use the Olink platform, which uses the proximity extension assay. So in short, kind of how this works is that it acts as two separate binding antibodies that target your protein of interest, which is conjugated to an oligomer. And when both of the antibodies bind to the protein, the oligomers recognize each other and then can extend. And from there, it's your simple PCR, so you can measure the amount of fluorescence from each of these detected proteins. So then we take that data and we do what's next. So first we looked at the known AD biomarkers adjusting for age, sex, years of education, BMI, smoking, hypertension, diabetes, and dyslipidemia, which is quite a lot of things here. And one of the important findings that we found is that ApoE4 is significantly associated with cerebrospinal fluid, A-beta-42, and SNAP25, but we didn't see this in the ApoE2, and we use ApoE3 as the reference group, where A-beta-42 is a form of amyloid beta, a pathological hallmark for AD diagnosis, and SNAP25 is a marker for neuronal death. So we see that, on a side note, we also see that CSF3 is only significantly associated with ApoE4 and not with ApoE3, again, using ApoE3 as our reference group since it is the most common in the population. And to note that CSF3 has been used in clinical trials with varying efficacies. And we think this is perhaps due to the fact that CSF3 in clinical trials might be used better if they were targeted to people with ApoE4 if they stratified by ApoE genotype, since they have varying efficacies. So here are the significant AD biomarkers and some select inflammatory markers graphed out to show the directionality between each of the ApoE genotypes. We then associate the known AD biomarkers with our inflammatory markers that we ran using Olink. And we see the consistent association of the same inflammatory molecules that are known in these known ApoE biomarkers. So next, with all this data, we wanted to see if we could create a prediction model with the addition of new inflammatory molecules along with the known AD biomarkers and epidemiological and clinical data that we tested for. The reason we wanted the patients to have at least 2 follow-up visits was to find that if any of the patients converted to mild cognitive impairment. And this is kind of what we found. There was no significant association between older age at the baseline visit and increased risk for CDR conversion after adjusting for all of the adjustments you see over here above. There were no suggestive or significant associations with CDR conversion observed other than with the epidemiological characteristics of age and possibly ApoE. So the ApoE genotype did show a trend at 0.031. So among inflammatory molecules with AD biomarkers, statistically significant associations with CDR conversion were observed with A-beta-42 as shown here. Should actually just close that so I can see that. A-beta-42, and then we also see it here with neurofibrillary light chain, and some of the inflammatory molecules. We see REG4, MICA-B, and then MICA, and SCGB3A2. And so taking these all into consideration, we wanted to create a prediction model for CDR conversion. And so what I want to emphasize here is that just with age, A-beta-42, NFL, we were able to get predictions kind of a little bit better than just a coin flip. And so, however, with the addition of REG4, one of these new markers that we probed for in our inflammatory marker panel, we were able to increase our prediction percentage to around 80%. So our C index was at a 0.62 with a moderate improvement of 0.063 compared to an age-only model, where age, again, is our most significant risk for developing Alzheimer's disease. So in summary, ApoE differentially expresses inflammatory molecules. ApoE4 is significantly associated with CSF, A-beta-42, and SNAP25, known AD biomarkers, consistent with differential regulation of certain inflammatory markers associated with known AD biomarkers suggests a chronological order of deterioration with age and AD. There's a significant association between older age, A-beta-42, NFL, and increased risk for CDR conversion. REG4 is differentially regulated after adjusting for clinical diagnostic diagnostics and associated with CDR conversion. And we were able to predict CDR conversion with up to an 80+% accuracy with the addition of Reg4 to known AD biomarkers in age. And if you were wondering what Reg4 is, Reg4 stands for Regenerating Family Member 4, and it's often produced by the pancreas and the of many external or peripheral organs, such as the small intestine. So it's one of the things that we are looking to kind of explore later on, instead of using cerebral spinal fluid, possibly looking into its association between the cerebral spinal fluid, these biomarkers, and in plasma. So I would like to thank all the co-authors on this paper, my thesis advisory committee, our U19 funding source, the Mayo Clinic Study of Aging and Patients and Partners, families, and the labs I've been in. And so here is a QR code to our newest publication that just came out less than a month ago. And we've also created an interactive graphical database if you're interested in graphing and kind of seeing how that data all works together. So thank you for your time.
That was great. Thank you, Frances. The final presentation of this session is from Ana Luisa Coutinho from University of Maryland. She'll be talking about performance of volume of distribution prediction methods for lipophilic drugs with uncertainty in log P values.
Hello, everyone. Can everyone see my slides?
Yes, we can.
Yes.
Thank you.
Great. So yeah, thank you for having me. My name is Anna, and I'm a pharmacist, and I just completed my PhD work with Dr. James Pauly at University of Maryland School of Pharmacy in Baltimore. And my presentation today is about the relative performance of volume distribution prediction methods for lipophilic drugs with uncertainty in log p-value. And this work has generated two publications, and it was performed with collaborators at the FDA, University of Florida, the Fraunhofer Institute of Translational Medicine and Pharmacology. So a little bit of a background. Oops. Volume distribution is a pharmacokinetic parameter that describes the apparent volume that the drug distributes within the body. It's not a real physiological volume. And the simplest way of describing volume of distribution is is the drug concentration after intravenous bolus injection. So that volume that divided, that dissolves all that amount of drug given, it is the apparent volume distribution. However, volume distribution is a much more complex parameter that's modulated by the ratio of drug present in different tissues in our body. The other parameter important in this research is log P. This is a drug physical-chemical property that describes the relative affinity of a drug to a lipophilic environment. So log P is the log of the partition coefficient in a binary system. So if you think about drug dissolved in water and drug dissolved in octanol, which is a very lipophilic solvent, think about that ratio of how the drug distributes within those two solvents. So that's what log P describes. And it is also the main drug property that affects the volume of distribution of a drug. So drug distribution, is modulated by the drug present in the blood, in the extracellular fluid, and also in the many tissues in our body. It's the physiologically based prediction methods used here predict volume distribution by, in part, considering the mechanisms behind the partition of the drug in these different compartments in the body. Depending on the drug molecule physical-chemical properties, the molecules may stay in the blood, but they also may spread to the extracellular fluid, but it also may spread to our different tissues. The drug properties also modulate if the drug will bind to proteins, which will also affect how they move around the body. So the drug partitioning, which is partitioning between these compartments, is a function of log P and other physical-chemical properties, but log P is the most important one. And if you think in general, volume distribution, it's a function of the drug partitioning and also the volume of the tissue where the drug's partitioning to. Prediction of drug pharmacokinetics in humans is critically important in early drug discovery and development. Prior to human studies, knowledge of the drug disposition in the body is useful to inform decisions on drug safety and efficacy, also to decide on first-in-human dosing, and also to decide on a dosing regimen, designed for studies. So our research addresses an important pharmacokinetic problem, which is how to estimate apparent volume distribution of a drug in the human body at steady state before you do clinical studies in humans. And the motivation of to study volume of distribution prediction methods was because we were conducting ourselves a PK study using itraconazole tablets. And itraconazole is a drug with high lipophilicity. And before, we were aiming to predict itraconazole plasma concentrations, but we observed a big change on volume of distribution values using different log P values for itraconazole. And so we decided to investigate the impact of log P values on volume distribution predictions. Volume distribution predictions are important, as I mentioned, in first-in-human dosing and pediatric dosing predictions, and also in therapeutic drug monitoring. And we know the log P is the most influential parameter in determining tissue to plasma partition coefficient and volume distribution predictions. So our ultimate goal was to assess the impact of log P in volume distribution predictions using 6 physiologically based prediction methods. The 6 physiologically based methods investigated here were the Oitoiser method, 2 ways of calculating the Rogers and Roland method. We also used the GastroPlus method, which is embedded in the GastroPlus software, which is a variation of the Rogers and Roland method. We also looked into the Kozak-Nagar method, and also in this new tissue composition-based model called TCM-New that was developed last fall. We conduct a sensitivity analysis and a prediction error analysis. In objective 1, we were concerned that the sensitivity analysis over a range of log P values for specific drugs, and the objective 2 was to concern prediction error analysis using specific log P values. And we worked with griseofulvin, itraconazole, posaconazole, and isovuconazole. Those 4 drugs are considered highly lipophilic because they have experimental log P values that are greater than 3. So here we have 3 sources of log P. So the accuracy and even availability of log P values is an issue, as there is a lack of experimentally determined log P values in the literature. Here in this table, the ADMET predictor log P values are in silico. So this is in silico values for log P for those 4 drugs. The literature log P that I have here is a mix of in silico methods, some experimental methods. And HPLC-based log P is log P that was calculated using a high-performance liquid chromatography method that was done in our lab. So this is 100% experimental. So there is we also questioned the accuracy of reported log P values in the literature. Others have indicated that highly lipophilic drugs often do not have experimentally log P measured. We aim to determine how variation, how log P will change if I use this value versus this value versus this value for those 4 drugs, and see how sensitive are the changes in volume of distribution prediction depending on the log P value that we're using. So here, this figure shows the volume distribution predictions of griseofulvin, itraconazole, posaconazole, and isovuconazole. So here we have volume distribution over log P value. And the green line shows the reported volume of distribution value observed in human clinical studies. And the vertical dashed lines denote the low and upper limits of reported log P values. And the horizontal dashed lines denote the twofold error values from the literature volume distribution, the green line. As you can see, methods such as GastroPlus and the Kozak-Nagar methods show a substantial increase in log P values increasing in volume distribution values as log P values increased, as you see in the orange and the black curves. You see they increase quite a bit as the log P values increase. However, when we use the Rogers and Roland methods, which are widely used, those are very famous methods. When we use the Rogers and Roland methods, we see a much steeper increase in the volume distribution values. Actually, some of the values go 80 times what was actually observed in humans. So as you can see, Rogers and Roland does not seem to be a good method to be used in highly lipophilic drugs. We also looked into prediction error calculation. So we looked into average fold error and absolute fold error. So here, just think about AFE looks at bias of the prediction and AAFE indicates precision. So the acceptable criteria will be a twofold error. So if you look at AFE and AAFE, that's within 0.5 and 2 times observed value, is a successful criteria. So if I looked at all sources of log P at different methods, you can see on the cells in pink shows that that the criteria, the values are outside the criteria, and the green cells are the values are in the acceptable criteria. So as you can see here, at low log P values, which is in silico methods, ADMET predictor, the GastroPlus method is the best. And interestingly, those are both the GastroPlus method and the ADMET predictor in silico log P values are both come from the same company. Their predictions all from the same company. They work very well. But if you use log P values from other sources, GastroPlus wouldn't be a good method. Rogers ruler wouldn't be a good method. So overall, TCM-New is a method developed by researchers last fall and shown to be a great prediction method regardless of the source of log P, which is very interesting. It's great to see that regardless of the values they're using, TCM-New would be a good option. When combining all prediction volume distributions with the 3 log P sources for all the 4 drugs, TCM-New was the most accurate method. Even though AFE and AAFE here is the same as the Oitoiser, when you look at each prediction for each drug at each log P value for each source, TCM-New is the best method. It's the most accurate, closer to 1. And so our findings here show that Rogers and Rowland's not a good method, even though it's widely used, not a good method for highly lipophilic drugs. And so these findings are according with previous research, we know showing that Oitoiser and TCM-New are great methods for highly lipophilic drugs. And Rogers and Rowland should not be used with drugs with log P greater than 3. So overall, TCM-New method, developed last fall was the best method for highly lipophilic drugs. It's suggesting that, using this blood plasma ratio was a favorable surrogate to show partitioning of the drug between plasma and tissues. And Rogers and Rowland dramatically will predict volume distribution of lipophilic drugs. GastroPlus and Kazakh-Nagar showed improvement in the volume distribution prediction errors compared to Rogers and Rowland, but it's still the best method was TCM-New. So we would suggest that researchers, sponsors, companies look at, take care in identifying and apply log P methods and values in estimating volume distribution in drug development and regulatory applications, because that will have a big impact on those predictions for future studies. Thank you so much. This is my presentation. I'm looking forward for any questions.
Thank you, Anna. That was great. Any questions for any of the speakers? Please feel free to put stuff in the chat. Alonso, maybe I can ask you to speak first about your work. Just from a learning point of view, as I said, I don't have the technical expertise to ask about some of the specifics, but I wonder whether you could reflect on, as you described your work and where it's at with, some, animal implantation and kind of early signs of success. What's the general rule of thumb in terms of the translation to actual clinical care, where when you're already have reached the point where you've reached, which is hard enough to get to? Is the idea like you'll be doing trials in humans in a year, trials in humans in 10 years, you may never get to trials in humans? How does that process work, and how does it happen from within academia versus the move to industry? I just think a lot of the people here might find that your reflections on that interesting.
Thank you, Dr. Rouse. So to my understanding, the FDA typically wants about a month of a month-long study. So currently we're doing feasibility. As I presented, we did a feasibility test to make sure that our protocol worked and that everything functioned as it should, and we actually produced ultrafiltration similar to the functions of the kidney and so on and so forth. But to my understanding, about a month long study in animals is looked upon favorably to move on to clinical trials. And um, as I previously mentioned, for our particular studies, we have two components, one of which is the filtration unit, and I work with the cell-based component. The filtration unit is actually much further along in its capacity to be implemented into a clinical trial. And so we are currently working on the cell-based component to enhance, the cell capacity. But we intend to have month-long data in the near future so that we can move to the next step.
Amazing. Congratulations. Anna, similar, I'm in the same boat and no one else is asking questions, but I'm curious. So without the technical expertise, I'm curious, in my world, the sort of translation of a clinical measure or some other tool that's used in research or in medicine, there's often a lot of kind of volleying back and forth between what's best, what's worse. And it's interesting that you've evaluated now these 6 different methods for these lipophilic evaluations. And are like, how readily will the pharmacokinetic community move from one to the other? Like, is it to how many different types of studies like yours will need to be done before people are convinced that, oh, you know what, we really should be using this newer method, not the older method? Are there licensure issues? What leads a company to be using one versus the other? Just kind of a little bit more big picture.
Yes, no, that's a great question. I always have this issue too that some of the softwares already come with a predetermined volume distribution prediction model and may not be the best for your drug. So it's kind of a challenge when you want to use one method, but the software that your company has a license for may not be using that specific method. But I think it's important to evaluate different methods, and there won't be one that it's always going to be right for a specific drug. But I think you should be analyzing different prediction methods before, of course, you start your first in-human dosing, because you want to get that dosing regimen correct. You want to get that dose correct and dose regimen correct, so you don't have a clinical trial that's not actually investigating. It's not going to give you, it's going to give you important information for the upcoming trials. So I would look at different methods, really look to see if the prediction makes sense too, because you're going to have, industry usually has a little bit of animal data to compare to. So I think if you have an understanding also of the physical chemical properties of your drug and look at different methods, you can come up with a first in human dosing, first in human regimen that makes sense. But it is a challenge when you have the softwares have very embedded prediction methods that you may not want to use. It's kind of annoying that part of different softwares may want to do their own calculations. But yeah, but just looking at different methods. Don't just use one prediction method. Look at all of them.
Got it. And just a last question for Francis. And one is a point of clarification, because I didn't hear whether the patients that you were studying, if they were pre-symptomatic or already symptomatic in terms of the sort of transition to cognitive decline. And I'm wondering, kind of just really more interested in next steps for your work. I mean, in terms of validating the biomarker predictions and larger patient populations over longer periods of time, and sort of where you'll go next.
Yeah, so to qualify for the study, you have to be cognitively normal. And so that's the CDR rating that we have for all of our patients. And so a lot of these patients have now passed away. And so we have their brains to kind of clarify whether or not they like which disease that they'd be classified under, which is why we do require so many follow-ups to be included in the Mayo Clinic Study of Aging. We do have some collaborations with WashU, and we are trying to validate in larger patient populations, since the Mayo Clinic Study of Aging is mostly Caucasian and very familial. And so, unfortunately, but one of the things that we were able to parse out that I don't know if will be of interest to Alyssa is that REG4 is highly upregulated in obesity and diabetes. And so with kind of decreases of those inflammatory factors that are onset by that. We could see like longitudinal potentially predictions that would shift based off of this model system. And so kind of where we're going with it is that like if we were able to kind of correlate that with some of the plasma studies that we do have corresponding plasma to see like what's going on. Hi, Alyssa. That we could probably implement this in Quest or some LabCorp or something, and to one determine whether or not, like, let's say when the FDA lecanemab has been effective in kind of bringing out that potential like inflammatory factors that lead to Alzheimer's, or actively be able to kind of take these existing labs and add an additional marker so we can predict active participation.
That's great. Well, congratulations to all three of you on some really nice work. I'm going to hand the baton back to Molly for session 4.
Thank you very much. And thank you all for hanging on for the last panel, which has some really fantastic work. We saved some of the best for last. So first up is Yu-Hong Mo, talking about composite endpoints with win statistics in group sequential clinical trials.
Hello, can everyone see my screen?
Yes.
Okay, great. Yeah. Okay, sorry for that. Hello, everyone. My name is Yun Han, and I'm a first-year PhD student in biostatistics at Yale School of Public Health. And I'm currently working with my faculty advisor, Dr. Yuan Huang. It is my great honor to join the Yale-Mayo CERSI program this year. And my research is testing composite endpoints with win ratio method in group sequential clinical trials. In today's presentation, I will first introduce the win ratio method and the group sequential design in clinical trials. And then I will discuss the research objectives of my project. And then I will give a brief summary to the talk. First, let us have a look at the win ratio method. In clinical trials, especially cardiovascular clinical trials, composite endpoints are widely used. For example, we may consider like time to death and time to hospitalization. So we have like more than one outcome of interest for the primary analysis. The traditional tool is to use the time to first event analysis. For example, in our time to death and time to hospitalization setting, for patient A, if both hospitalization events and death events are observed, then the first event will be recorded as the final measure. As we know, hospitalization, of course, happens before death event. It is pretty strange for using the time to first event analysis here, since we know death events are more important than hospitalization events, but you are always recording the hospitalization event if you have both. To address such differential clinical importance of endpoints, the win ratio method was introduced in 2012. The win ratio method is based on pairwise comparison with the hierarchy. Let's have a look at the comparison between two participants, I and J. We will first examine their survival difference. That is, if I lives longer than J, we will conclude that I wins the pairwise comparison. And if J lives longer than I, we will conclude that I loses this pairwise comparison. If we can't make a conclusion, maybe like both participants do not have death events observed, then we will move on to the hospitalization difference. Similarly, we can claim I win or lose in this comparison. If there is still no conclusion, we will claim the comparison has a tie here for participants I and J. And then we can calculate the winning ratio, RW, which is equal to the number of wins obtained by the treatment group participants divided by the number of losses obtained by the treatment group participants. With the RW, if RW is significantly larger than 1, then we may conclude that the treatment is better than the control simply because the treatment group participants are winning more than losing. There are multiple approaches available for testing the null hypothesis, that is, whether there is treatment effect or not. For example, we may use the FS test statistics with the comparison results of all pairs of participants we can construct these statistics, which is asymptotically normal under the U statistics theory to test the null hypothesis. And with the advantages of addressing differential clinical importance of multiple endpoints with its hierarchical structure, the win ratio method is now included in the 2022 FDA guidance on multiple endpoints in clinical trials as one of the recommended methods. And now let's have a look at the group sequential design. In the standard clinical trial design, we recruit participants, we perform the treatment and control interventions, and then we follow them up. At the end of the study, we have all the data collected, and then we analyze the data once to see if we want to reject or accept the null hypothesis. But with the group sequential design, we will have pre-specified interim data analysis. That is, instead of having just one test statistics day, we will have like day 1, day 2, all the way to day k. At the time point, at the x-axis can be regarded as the calendar time as well. So at the first interim analysis, we have test statistics day 1. If day 1 is very large, that is strongly significant above the red line, we can conclude our treatment is already better than the control. So we will reject the null hypothesis at the first interim analysis. On the other hand, if day 1 is strongly non-significant, that is very small, below the blue line, we will have to say that there is just little chance for us to reject the null hypothesis, even if we finish the whole study. So we have to accept the null hypothesis here, claim that the treatment is like similar to the control. At this time point, we can give early stop if possible. The red line and blue line will be like going together at the last study, because at the last data analysis, we will have to make the decision whether we are gonna reject or accept the hypothesis. We won't have the chance to progress the study anymore. So the boundary here, the red line and blue lines are pretty important. We need to calculate them regarding the overall type 1 error rate, because we're looking at the same group of participants or like the data set, the cumulative data set for multiple times. And the advantages of the group sequential design is that we can make earlier decisions. If we can conclude the treatment is effective at the first analysis, we can terminate the study and save money, save time, make the good treatment available to patients earlier. Also, we may ended up recruiting fewer participants. And the third thing is that we may also be able to stop the failing trials earlier, so that we can, avoid exposing more participants to potential risks for no reason. With the advantages of the win ratio method and the group sequential design, it is very natural for us to consider if we can use the win ratio method to test the composite endpoints while having the group sequential design in a clinical trial. This makes 2 challenges for us. First is that in the analysis stage, we need to calculate the boundaries, that is the red and blue lines in figure 2, so that we can maintain the nominal significance level. And on the design stage, we need to calculate the sample size so we can make a good budget plan and maintain the desired power. So in this research, I will try to provide computation tools for the boundary and sample size calculation. I will first try to provide analytic solution that is the closed form formulas if possible. But based on my current progress, I will have to see if we are using the win ratio method. It is likely that the analytic solution exists for only some specific scenarios. So simulation-based approaches are necessary. And for the simulation-based part, I will provide R script or R packages for programming like calculation for the boundaries and sample size. And also I will try to provide maybe a no-code version with the R-Shiny app so that the sample size calculation can be done without programming. This might be helpful for like practitioners if they want to, have a look at the sample size before discussion with biostatisticians. And also, we will provide some designing and analyzing examples to demonstrate the use of the developed tools. And I will also offer two tips and tutorials on using the win ratio method together with the group sequential design in practice, since there are like many details in combining two interesting methods together. How can they work like best with each other? If I have time, I will also address other closely related win statistics, such as the win odds, win probability, and win loss. They're very similar to the win ratio. They all have the hierarchical structure. They all features the pairwise comparison, but they all have their own features. For example, the win odds might be better at addressing the ties in the pairwise comparison results than the win ratio. So I want to try to provide a guidance on, under what scenarios which like win statistics might be the best to use. And as a brief summary, in this research project, I will try to address practice and implementation details and challenges when we want to use both win ratio method and the group sequential design in clinical trials. And here are some key references. Thank you for your time, and questions and comments are appreciated. I believe it will be saved to the Q&A session. So thank you very much.
Yes, exactly. Thank you so much. That was fantastic. Next up, we will have Harry Dornberg talking about the impact of boxed warnings on prescription patterns, among other outcomes.
Thank you. Let me see if I can change the screen.
Looks good.
All set? Oh, thank you guys. I know it's been a long day, a lot of many talks in a row. And so I appreciate you guys being here. I'm talking about something maybe slightly different than what's come before. Looking at FDA safety actions in the post-market period, I'm really trying to get a sense of a very simple question of, do these box warnings and post-market safety actions have the effect we think they have? So just a little bit of background. So as many of you guys on this call know, the FDA has to regulate drugs, not just in the pre-approval, pre-market stage, but also after drugs get approved, because clinical trials that support approval can only be so long and enroll so many patients. So you can't have discovered all of the adverse events that and effects of a drug at the time of approval. So there needs to be a mechanism by which the FDA can go back and communicate to providers and prescribers that there's new evidence about safety events or new risk-benefit profiles for drugs. So the FDA has several mechanisms by which they do this. It's streamlined a little bit now in the past since 2010, but obviously they can withdraw medications, they can issue some advisories, but the main methods that the FDA communicates risk in the post-market time frame are through drug safety communications and box warnings, also called used to be called black box warnings. And the use of these box warnings as they become the major type of safety communication has likely increased over the past couple of decades. The first one was put out in 1979, and since then there have been many, many more. So what do these box warnings look like? So I think the physicians and other prescribers are familiar with them and definitely orally kind of understand what a box warning is, but they can kind of fall into several different categories. The most frequent and one that people think about immediately is this increased harm, particularly either in a general population or in a subset of population. So a good example is, and sort of the classic example is within children and adolescents for antidepressants, an increased risk of suicidal ideation and thoughts. That was kind of a side effect of these medications that was only discovered in the post-market period, and for which the FDA added a box warning afterwards. But these box warnings also have other content, the drug-drug interactions, that weren't discovered in the pre-market period, or just increased patient monitoring, like if it becomes best practice to monitor someone's liver function tests and enzymes in the future when they're on these medications. So all in all, we take for granted that these drug safety communications work in the way that they're supposed to, that prescribers will decrease the prescription in certain populations where it's deemed less safe, or it will continue monitoring, or when that's called for. But that is pretty much what it just is. It's an assumption. We actually don't really have a good sense across the board whether these work in the way that they're intended. So some studies look at these box warnings individually, included from the Ross group that looked at fluoroquinolones for like acute sinusitis and acute infections, which the FDA issued a box warning against a while ago, to see whether the prescription patterns changed. And often these have had pretty mixed results. Some papers show positive effects as in the box warnings had the effect that were intended. But many studies show that there actually were mixed results or opposite results or nothing at all. So one of the things that we want to do is look at this body of literature as a whole and look at all the studies that evaluated the effect of box warnings on prescription patterns, health behaviors, and any dimension like that. There are 2 existing studies that do similar things, one in 2010 that looked at sort of all FDA regulatory actions. Since 2010, though, there have been 40 box warnings that the FDA has put forth, and many other studies. So we want to take another look again at these effects. The study from 2020 was pretty broad. It was non-FDA, and including the FDA, but it would include more global regulatory agencies. So pretty simple systematic review, looking through a bunch of sources in a particular time frame. We wanted just original research, so no viewpoints, editorials, and we also wanted to exclude like medical devices or advisories from abroad, non-FDA entities, and do a systematic review in that way. So right now we're still trying to optimize the search. As in systematic review, you want to cast a wide enough net that you include all the studies that you want, but are narrow enough that you don't have to trove through tens of thousands of irrelevant papers. And the original searches we've done have been difficult because there's a lot of literature on labeling for things like cigarettes and food, which is understandable why it gets caught in these searches, but is ultimately not what we're looking for. We're trying to figure out the best search method is still something we're working on. And then once we get the abstracts and the full text, we want to do some basic categorization, but more importantly, we want to look at, again, these box warnings on the actual health behaviors. And then we can kind of grade the value of the evidence and the strength of the evidence as we do it. Thank you, guys.
That was great. Thank you so much. And best of luck as you continue working on it. I know systematic reviews are really tough, and putting together your search strategy is a tough part of it. And finally, but definitely not least, Karthik Chetlapalli is going to talk about the portrayal of efficacy and safety in FDA press releases.
Alrighty. Hello, everyone. Can you hear me?
Yes, perfect.
Alrighty, guys. So I'm going to round this out with a kind of a similar kind of topic, kind of just tying into what Harry's been talking about in terms of the portrayal and communication of efficacy and risk information. Now, we all know that the FDA is an independent regulator that regulates the statements made by drug companies in terms of the efficacy and risk of the drugs. As you can see on the right-hand side, here are some examples of some of the advertisements we'll come across where certain companies will make claims regarding the efficacy of their drugs and advertise them accordingly. And so the FDA has a key role in regulating what companies can say. At the same time, the FDA as a public entity, the government entity, also puts out communications in the form of press releases. And sometimes these press releases can be a bit controversial, especially for controversial approval. As you can see on the left-hand side, this Alzheimer's press release got a lot of scrutiny. So going from there, it makes sense to just take a brief review on the types of regulations that exist that the FDA use to judge promotional advertising for prescription drugs. It really boils down to two main things. The first thing being the information that drug companies are allowed to put out. They're not allowed to be misleading with respect to side effects, the actual effectiveness of the drug, and the contraindications. Similarly, they need to be able to be promotional. They need to also be able to present a fair balance between information about this drug's efficacy, how well it performs, how well it helps patients, and its safety and risk. What are the drawbacks? What are the side effects? What are the things that patients need to look out for, including black box warnings, as Harry was talking about. In addition to that, in recent years, the FDA has put out guidances specifically for drug companies, and it's acknowledged the importance of quantitative language, actually quantifying the risk, quantifying the efficacy, and has cited numerous, research studies that have indicated that it has increased the consumer's ability, patient's abilities, doctor's abilities to form an accurate perception of a drug. So now we take these two things together. We take that the FDA is both a regulator of drug companies and what they can put out there regarding their drug, and as an entity that puts out information themselves in the form of press releases. Does the FDA follow their own rules? Is the information they're portraying in accordance with their guidances and the regulation out there already? So some more specific questions we wanted to ask is, looking at FDA press releases, do they present any vague information? Do they have that same fair balance between safety and efficacy information? And do they effectively quantify the level of risk, the level of effectiveness, using fair comparison to placebo groups, using actual numbers, percentages, and so on and so forth. So these are the three main things we wanted to look at in our study. And I wanted to really focus on the impact that this has on the perception and the relationships patients have with their physicians when patients are making a decision whether or not to choose a drug, whether or not to say yes when their physician offers them a treatment option. This is really critical when we think about the decisions that we make every single day in the hospital, in the lab, and so on and so forth. Let's get into some methods of how we went on exploring FDA press releases. Just to kind of a framework of our study, this was a descriptive, retrospective study of FDA and drug-sponsored press releases. So they were matched from 2018 to 2023. The way we did that was we went to the FDA website, we accumulated all of the FDA press releases for specifically novel drug approvals, and we matched that to press releases that were issued to newswire websites from the drug sponsors themselves. Then what we were able to do was we were able to individually read through all these articles, which I was able to do, and kind of form our initial kind of impression of the common types of statements made. And from there, we were able to code statements into distinct and discrete categories. From there, we also wanted to look at specific regulations, and we added on additional subdomains that we were able to code statements in. And finally, when we extract these statements, we were able to compare the two between the FDA, drug sponsor, and the guidances and see where are places that are mismatching, where are places that are misrepresenting information, and so on and so forth. So let's take a look. On the left-hand side, we have an example of an FDA press release. On the right-hand side, we have an example of a drug-sponsored press release. And this is also for that same approval for an Alzheimer's drug. So reading these two articles, my approach and the approach that we took in this study was, we would then break them down individually to common things that would occur, common statements that were made specifically. On the left-hand side, you'll see the FDA press releases tend to format, they acknowledge themselves, they use a template, and it tended to include this information. In general, efficacy, safety, required trials, and regulatory. In that order, you would see that trend in every single FDA press release. On the right-hand side, you'll see a drug-sponsored press release would tend to be longer, tend to include a little bit more promotional language, but also they included something very particular, dedicated safety and efficacy section. And that's something I'll talk a little bit more about later. That was adopted from promotional labeling requirements and drug labeling requirements overall that they included in their press releases. And it's something important to mention as well. So now, just what are the key additional statements we wanted to investigate when we're looking at these press releases? I mentioned before all those guidances. So based off those guidances and those regulations, I wanted to kind of really drill down on specific areas. The first one being a portrayal of risk and quantifying that risk. And that's based on the guidances you can see on the right. Additionally, I wanted to look about statements made about the drug's mechanisms. There's been a lot of controversy, specifically in the Alzheimer's realm, about statements relating to a drug's actual mechanism action. So I took an extra look at that in statements made in best press releases and drug sponsors. Same thing for quantitative efficacy. The exact percentage of improvement of one drug over the placebo group. Statements made in that regard were also coded. In addition, characterization of the clinical trial, whether or not they were actually saying it's a single arm, an open label, a randomized study. These are the statements I also took a closer look into. And finally, side effects. What are the side effects of the drug, and whether or not the press release mentions the side effects or all of the side effects that are relevant. So just to summarize, the main methods become these broad categories that first parse the statements into, then more subdomains, and then finally those last subdomains are the ones that are rooted in guidances I just went through. So let's go through some results. Just to take a step back, just if we don't really know what's going on with these press releases in the first place, what's the purpose, we want to see just the general trends. So looking at that here, I found that in general, FDA press releases are released at a certain percentage of the actual number of actual novel drug approvals. Every year, if there's 50 novel drug approvals, maybe 20, maybe 10. And as you can see, the number of press releases issued for novel drug approvals has been declining over the years. And this may be due to a recent kind of increase in uptick in these short form kind of announcements called roundups, but that's a whole topic for another presentation. Some other trends I would like to highlight, year over year, you've seen the decline. You also see a representation of fast track and priority approvals significantly or substantially in these press releases. In addition, you see a good representation of hematology and oncology approval in these press releases. Now let's get to the meat of the project. The meat of the project really found, where were the areas that there was misleading information or an imbalance of information, or where things were a little bit vague, where things could be better communicated. On the left-hand side, you'll see the biggest one was quantitative risk, actually quantifying how often a side effect happened compared to the placebo. The FDA had 99 instances of this where they did not mention the quantitative risk of a drug. The sponsors did a majority of the time. There's only a few times they did not mention it. And that's, like I mentioned before, due to that additional dedicated safety and efficacy section. That's the main finding here. As you can see, there are also other subdomains where they still had mismatches, but that's the main one I want to focus on. And just some further characterization of these mismatches, you can see sometimes you'll see statements made such as for quantitative risk where the FDA may say common side effect is, and give no quantification. They're using a very basic qualitative description of the risk. On the right-hand side, you'll see that the sponsor tended to use quantitative language. They actually quantify if it happens in 20%, 40%, 90% of the patient population in the clinical trial. Similarly, you'll see areas where there's vagueness in terms of description of the mechanism. You'll see vagueness in terms of description of actual clinical trial outcomes, the characterization of the clinical trial, and the side effects. Sometimes there would be areas where a side effect was dropped or not even mentioned in the first place. So finally, I just want to end with a takeaway. When conveying information about risk, why does this all matter? Why do public communications really matter? I just wanted to present a scenario. For a patient that reads 2 articles, they read one from the drug sponsor and one from the FDA. One says the common side effects include, the other one says 40% of patients experience nausea. The patient may not know the difference between the two, but let's say they read another 2 articles. One of them says common side effects include neutropenia. The other one says 90% of patients experience neutropenia. And so for that, the patient now has a question mark. How common is common? What does that actually mean? 90% is pretty significant. 40%, maybe not so much. So this vagueness is important to take into account. And that's really what I take into account when I really at the end of this day, at the end of the study, that's what I want to take away from this. And so finally, just to final remarks, the number of FDA press releases have been decreasing year over year. Oncology drugs are often represented, but mainly quantitative language around safety has been lacking year over year, and therefore FDA templates should include a dedicated safety and efficacy section. Thank you so much.
That was terrific. I'm really excited about all three of these, and I have a million questions, but I'm going to open it up to the audience to see if anyone else has one first. So I have a question that kind of goes to both Harry and Karthik. And my question is about who is the FDA's audience, or who is the audience for like the black box warnings or these press releases, right? So medicine is a really strange field where the person who makes the decision is not the end user and it's not the purchaser, right? So, and I've talked to physicians and said, hey, you know, the opioid label says X, and they're like, the what now? So I'm not convinced that lots of physicians like know about these things or read about these things. So I would love to hear from both of you, your thoughts on the audience, who they should be targeting it to, and like what the mechanism could be for these things to have better effect. Karthik, do you want to go first?
Yeah, so I actually looked into so the FDA has a website where it basically went through every single announcement they make, and it gives the actual audience and the protocol that they go through for that announcement. And so for FDA press releases, the audience is news organizations, but also the public. So it's kind of vague in that sense, but it's written in a language that's supposed to be generally like understandable by the public. That includes patients and providers. But at the same time, the drug sponsor press releases that they regulate are also meant to be absorbable and understandable by the public. So they that's really where the mismatch is. So these are intended for the public. I guess that's my best way of putting it. But I'll let Harry answer the rest of his part as well.
Yeah, I mean, I think you sort of hit the nail on the head. It's sometimes these warnings don't reach the intended audience because the intended audience is not exactly clear. I think that boxed warnings in particular are meant for prescribers. That it used to be on the actual drug label itself that people would have to pick up. That being said, I think a lot of boxed warnings make it into safety communication to the general public. And in fact, a lot of the studies that will probably be included look at patient perceptions of risk before and after. And I think a lot of my non-medical friends know the box warning about certain obstetric products, antidepressants that make their way into the public sphere of knowledge. But generally, among all box warnings, the hundreds that there are, I think, are mostly geared towards physicians and prescribers.
Just to add on like a small example, recently I was working with a patient, and they're very enthusiastic and they read up on everything. And they were asking me, oh, I know this drug is associated with cytokine release. What does that actually mean? Am I going to have cytokine release? And so, it just made me recognize for a second, there are people that read these things. And so it doesn't really matter what at least in my mind, the intended audience doesn't matter as much. It's more of the actual impact and like whether or not it's written in a way that is beneficial to those that end up reading it.
Well, sort of on that point, the actual content of the box warning is often pretty technical, like, cytokine release syndrome or differentiation syndrome. So I think specifically the box warnings itself is geared a little bit more towards physicians. Sometimes the announcements about the box warnings that get picked up by the media or sometimes actually put out by the FDA themselves, based off of the language, is more towards the general population.
I mean, it's a great point. And Karthik, when you were showing the two examples, it seemed like the sponsor's language was much more technical to a point where you would have to talk to your physician in most cases about what it actually meant. And sometimes I wonder if that's like the person talking really fast at the end of the ad so that you can't understand what they're saying, where they're trying to sort of change how that lands.
I wonder if I could comment. I routinely try to tackle this kind of language of using adjectives rather than numbers in the medical writing of my fellows and students and junior faculty, because they will say things like, is common. And of course, you made the perfect description of what is common. 40% common is 90% common. In some context, is 5% common. It all depends upon the context. So I urge them to try to capture this quantitatively. But then there's the alternative challenge that numerical literacy is not particularly high in the US. And sometimes something like is common is better understood than it is 80%, you know. So I guess I was asking in the chat if you feel comfortable with your research at this point to develop a best practices for press releases, because if you could craft a best practices, it would force you to wrestle with some of these issues and what the best way to communicate with the public is. And if you came up with such a thing, I can imagine academia, industry, and the FDA itself benefiting from those guidelines. Even if they had to violate it from time to time for a good reason, at least they would have a kind of best practices document. Are you going to be that ambitious?
I don't know if I'm that ambitious, but I'd like to redirect that to the FDA because they're already putting out guidances. I base this project on the guidances that they put out. So I think it's
but you had a compelling critique of some of the elements of those guidelines, and you actually did a good job highlighting strengths and weaknesses both of FDA and industry. So I'm wondering if you could, using your judgment as a scientist and your thought leader in this area, if you could give a stab at best practices. Maybe not alone, maybe you do it in partnership with some of the people on this call, but you could take a lead.
It'll become my thesis.
I guess I'm recommending it. I think that would be an amazing thing to do.
Agree. Other questions? Oh, I see a comment in the chat that AI could potentially be helpful in clarifying the warnings. So I just had sort of a comment for you, Hong, about the work that you were doing, and I really wanted to note how much effort you're putting into dissemination of the results, which I think is just fascinating. The Our Shiny app that will allow people to like actually use what you're doing to easily use it in their own work, which I think is just fantastic.
Great, thank you. I just hope, because sometimes like clinicians, they want to like conduct a clinical trial, and maybe they want to just have a quick estimation of the sample size before they get biostatisticians involved, so that maybe a no-code version could be better. And I think the underlying code will be very similar. It's just like we're using the Our Shiny app to help you like program it automatically in a like an easier way. So could be helpful if
Yeah,
Thank
the
you.
self-service idea of it is fantastic. So I really commend you on that piece of it.
Thank you so much.
Yeah. Well, if there are no other questions, I will give some very quick closing remarks. I just want to thank everyone for joining us today for the annual CERSI Scholar Virtual Research Seminar. It is really such a pleasure to hear all of these presentations from such a diverse group of scholars addressing so many different kinds of questions across the whole spectrum of what the FDA does and what regulatory scientists do. So thank you to everyone who joined today, folks from the FDA, our scholars and their mentors, who we really thank as well, and friends of the CERSIs as well. The Scholars Program is a really important part of our mission as the Yale-Mayo CERSI, where we're trying to develop future regulatory scientists and to support the science that they're doing. And you can see that the science that they're doing is really cutting edge. And answering essential questions that help the FDA in its work regulating, as we heard today, products that make up 25% of consumer dollar spend in the US. So last of all, a huge thank you to Michelle Bernabeo for her work to coordinate the program today. Thank you so much. And we look forward to seeing you all back here next year. Thanks, everyone. Have a great weekend.
Thank you.